Hello students, and welcome to lecture 9 on the circulatory system, sometimes called the cardiovascular system, which is basically just the heart and the blood vessels. And so we have the heart, the lungs also known as the pulmonary system. The lungs are also a part of the circulatory system because the main purpose of the circulatory system is to pump blood around the body. And what is the blood doing? The blood is delivering fresh oxygen to all the cells because all of the mitochondria in all of the cells need oxygen in order to produce energy through the breakdown by breaking down glucose and producing ATP as a substitute. Okay, so oxygen is consumed in that process. So first of all, we have to have some way to deliver fresh oxygen to every one of the cells in the body. The second thing is that every cell in the body produces carbon dioxide as a waste product and sends that carbon dioxide out into the blood where it is taken to the lungs and sent out into the air. Another waste product, which we'll talk about later, is ammonia, and ammonia is produced as a waste product resulting from the breakdown of amino acids, and the breakdown and recycling of nucleotides often generates ammonia as a toxic waste product. So the cells want to get rid of the carbon dioxide, they want to get rid of the ammonia, there are a few other things that they get rid of, and they simply dump it back into the bloodstream. Okay, so the cells of the body are kind of using the bloodstream the way medieval human villages used to use rivers, right? So you know that in primitive societies, the villages would be located near the river, and the people who were fortunate enough to live near the mouth of the river, the source of the river, would get the freshest water. Right, so they would get fresh water, and at the same time they would dump their garbage and their sewage into the same river. And so then the people who are downstream from that village will get less fresh water that is contaminated with more sewage and garbage. And then you keep on going downstream until you get to the mouth of the river that leads out into the ocean. And the people who are unfortunate enough to live there are drinking very dirty water that has very little fresh water in it and a lot of garbage. The same is true for the circulatory system. So the cells that are located nearest the heart, for the organs and things that are located nearest the heart, they get lots of fresh oxygen, and they proceed to dump their carbon dioxide and their ammonia and their other garbage into the bloodstream. The blood continues on to the next organ. The blood contains slightly less oxygen and more waste products and so on, until finally the blood gets back to the heart. It is completely depleted of oxygen. So we say that the blood is oxygen depleted by the time it gets back to the heart where it started. And it is filled with carbon dioxide and ammonia. The oxygen depleted carbon dioxide enriched blood is then sent to the lungs. The carbon dioxide will go out of the blood into the air when we exhale, and then more oxygen will come into the blood, and then the process begins again. Okay, so that's the purpose of the circulatory system, is really to deliver fresh oxygen to all of the cells in the body. Depending on where those cells are, the oxygen may not be that fresh. So you deliver fresh oxygen to all the cells in the body, and you remove the carbon dioxide and the ammonia from all the cells in the body. As we mentioned in the renal section, the renal discussion, the ammonia is sent to the liver where it is converted to something less toxic called urea. We'll talk about that in another lecture. And then the urea is filtered out of the blood by the kidneys. This is the renal system, so we'll talk about that when we discuss the renal system. But the oxygen and carbon dioxide are put into the blood or taken out of the blood at the lungs. And so the lungs are sometimes considered to be part of the circulatory system. So the entire thing together we call the cardiopulmonary circuit. Cardiopulmonary, cardio meaning the heart and the blood vessels, and then pulmonary meaning the lungs. So the heart and the lungs and the blood vessels together constitute the circulatory system. Today we're mainly going to be talking about the heart and the blood vessels because we've already had another lecture on the respiratory system, but just remember that the lungs are an important part of the circulatory system as well. Okay, so today what I'm going to talk about in this lecture is we'll talk about the heart itself. We'll talk about how the heart works. What are the different compartments of the heart? There are actually 4 compartments that are referred to as chambers, right? There's a left and a right side of the heart. The left and the right side each are composed of 2 chambers, so there are 4 chambers in total. And the different chambers of the heart have different functions, which we'll discuss. And then we'll talk about learning the names of the major blood vessels. And so we have, in the blood vessels are the tubes, the tubes that carry blood around. Those are the blood vessels. Now, arteries carry blood away from the heart and generally carry blood that is oxygen enriched. So that blood is filled with newly acquired oxygen that was picked up at the lungs. Veins usually carry blood back to the heart, and usually veins carry blood that is oxygen depleted. So it has already delivered all of the oxygen to all of its stops along the way, and now the blood is oxygen depleted and it's heading back to the heart and then back to the lungs to get new oxygen. So generally arteries carry oxygen enriched blood and veins carry oxygen depleted blood, but that is not always the case. There are 1 or 2 very notable exceptions to that rule when we get near the heart. So the actual official definition of an artery is a blood vessel that takes blood away from the heart. And the actual official definition of a vein is a blood vessel that takes blood back to the heart, regardless of whether it's oxygen enriched or oxygen depleted. And so we'll see where the exceptions to these rules are. Capillaries are very thin blood vessels that are made of only a single layer of simple squamous epithelium. And the reason why they're made of those cells is because they need to be thin. They need to be thin because the oxygen and carbon dioxide exchange between the blood supply and the cells is meant to take place in the capillaries. Right? So oxygen is never transferred from arteries to organs. Instead, the blood is transferred from arteries to capillaries, and once in the capillaries, the oxygen-enriched blood will deliver its oxygen to the organs. Right? So the organs are wrapped in what are called capillary beds or capillary networks, and that is where the oxygen carbon dioxide exchange takes place, in the capillaries. So arteries and veins are merely meant to transport blood around the body, whether it's oxygen enriched or oxygen depleted. And capillaries are designed for exchange of oxygen and carbon dioxide with the cells. Right? So the arteries and the veins have very thick walls, and oxygen and carbon dioxide cannot diffuse across those walls. And so those blood vessels are made only for transportation. The capillaries have very thin walls, and they are made for oxygen carbon dioxide exchange. So oxygen and carbon dioxide can diffuse across the walls of a capillary very easily, because those walls are very thin. They're made of only a single layer of simple squamous epithelium. Okay, then when we're discussing the blood vessels, we will talk about how blood pressure is measured. You know that when you go to the doctor's office, they put an inflatable rubber cuff around your arm just above the elbow. That's called a sphygmomanometer. And then they blow it up, and then they listen with another instrument called a stethoscope that they use to listen for the pulse. They're actually measuring the blood pressure in a special artery that's located in your arm just above your elbow called the brachial artery. Brachial is an anatomical term that means the upper arm, right? So if you take Biology 130, you'll learn all about the brachial artery and the brachia, the specific region of the human body. And so the brachial artery is what's used to measure blood pressure, and we will learn exactly how to do that and what the numbers mean. And then at the end of this lecture, we'll talk about the blood itself, because the blood is considered to be part of the circulatory system. And the blood is composed of what are called formed elements, the cells. And we have 2 types of cells. We have red blood cells and white blood cells. The red blood cells are called erythrocytes. The word erythro is a Greek word meaning red. And the white blood cells are technically called leukocytes. The word leuko is an ancient Greek word meaning white. So you've heard of the disease called leukemia. Leukemia means cancer of the white blood cells, and so the word leuko or leukocyte is contained in the word leukemia, for instance. So somebody who has leukemia is unfortunately, they have way too many white blood cells in their blood. Those white blood cells are actually part of the immune system, and those immune system cells start attacking other structures in your own body. So basically, leukemia is a tragic example of a person's own immune system having gone wild and start attacking their own tissues. Okay, but for now we're talking about transportation of oxygen. We're not talking about the immune system. The lecture right after this one is going to be on the immune system, so we will talk all about the white blood cells. We will talk all about the leukocytes when we talk about the immune system. For now, we're only interested in the erythrocytes, that are the red blood cells that carry the oxygen. So the blood is composed of formed elements, those are the cells, as well as the liquid portion, which is called the plasma. So the plasma is the liquid portion of the blood. Okay, now there is a measurement that doctors and nurses will do if you go to the hospital, a measurement. If they want to know how many blood cells are contained in your blood, they will measure something called the hematocrit. Right? The hematocrit is if the blood is made of liquid as well as cells, and so they're interested in knowing how many cells are there. And so what they do is they concentrate the cells, and then they measure what percentage of the blood volume is taken up by the cells. That is called the hematocrit. And that number is expressed as a percentage. The percentage is usually about 45%. Right? So the standard hematocrit for a healthy person is about 45%. That means that for a healthy person, about 45% of the blood is taken up by the cells. The other 65% is taken up by the plasma, the liquid. Okay, so that number is one that you need to know, that term. You need to know what this term means. The hematocrit is the percentage of the blood volume that is taken up by the blood cells, both the red and white cells. And it's a critical number because if your hematocrit is low, that means you're anemic. The word anemic, anemia, means that you have too few red blood cells. Okay, so then at the end of this lecture, we'll talk about the ABO blood groupings. You know that when you have a blood transplant, if you're having surgery, or if you've lost some blood because you were wounded or you were injured in an automobile accident or something like that, and you've lost blood, they need to put some blood into you. That blood is obtained from donors, so generous people go and they give their blood for free, using in the United States and a lot of other places in the world, Western Europe and so on. The agency that collects the blood is called the Red Cross. In Muslim countries, there's a branch of the Red Cross called the Red Crescent. They collect blood for donation. We used to have the Red Cross in Canada, but the Red Cross in Canada in the 1980s did something very bad, which was that they transplanted blood into people without checking to see if the HIV virus was present first. So a lot of people contracted HIV, AIDS, by getting blood transfusions in the early 80s. As a result of that, there were some big lawsuits, and the Canadian Red Cross went bankrupt essentially and disappeared. And now we have a new organization that's called the Canadian Blood Services, which is used to collect blood from donors, and of course they do check it for HIV and other diseases to make sure that something like that never happens again. But that's the reason why we don't have the Red Cross collecting our blood from donors in Canada. It's because of a sad, tragic event that happened in the early 1980s. All right, so you know that the people collect blood from donors and then they give it to people who need it, but in order to give it, you have to match the blood grouping. In other words, are you blood type A, blood type B, blood type AB, or blood type O? There are 4 different blood groups, and we'll explain how that works. And you can only donate, a donor you can only accept, if you're receiving the blood that somebody has donated, you can only accept blood from a compatible donor. And we'll explain what that means, because the immune system, if you take blood from somebody who's not a match, who does not have the same blood group as you do, your immune system will assume that these cells, these blood cells that have gotten into your body, they are invading organisms, invading microorganisms, and your own immune system will destroy them, will kill them and destroy them, which is the function of the immune system. The function of the human immune system, as well as the immune systems of other mammals, the function of the immune system is to destroy foreign cells that invade your body. And if you are blood type O and somebody puts blood type A into your body, your immune system will perceive those type A blood cells as foreign invaders, and your immune system will destroy them. So that would be pointless, wouldn't it? Okay, so you can only accept blood from a compatible donor, and that compatibility is based on the ABO blood grouping system. All right, let's talk about the heart. So the word cardio or cardiac always means the heart. The word myocardium refers to the heart muscle itself. All right, so as I said, arteries carry blood away from the heart, veins carry blood back to the heart, and capillaries are microscopic blood vessels that deliver oxygen to the tissues. They are made of a single layer of simple squamous epithelium, and there's quite a lot of them. So they kind of resemble a fishnet because they are kind of like a webbing rather than a tube. And they're sort of in the shape of a web because they're meant to wrap around all the areas of an organ or a tissue and deliver lots of oxygen-enriched blood to that tissue. Okay, so arteries bring blood to capillaries, and the capillaries deliver oxygen to the cells, and they take away the waste products, including ammonia and carbon dioxide. The capillaries then drain into the veins, and the veins go back to the heart. Okay, here in this diagram we can see capillaries, arteries, and veins. Okay, so here's an artery leaving the heart. Notice that it has very thick walls, and the walls are divided into 3 different layers. The central layer, the layer that faces on the inside, is called the endothelium. Okay, now here's something that you need to know. The inner surface of a blood vessel, either a vein or an artery, is made of squamous epithelium, usually simple squamous epithelium. However, when we're talking about the simple squamous epithelium that lines the inside of a blood vessel, we give it a special name, and that special name is endothelium. Endothelium, right? So the inner surface of a blood vessel is lined with simple squamous epithelium, but whenever we find simple squamous epithelium on the inside of a blood vessel, we give it a special name, and that special name is endothelium. Endothelium, right? So I could very well ask you that on a test. I probably will. Okay, so then outside the layer of endothelium, we have a layer of smooth muscle. Right? The layer of smooth muscle is to allow the blood vessels to contract or expand. Right? So if you squeeze the blood vessel, it will become narrow, and that is called vasoconstriction. To constrict means to make narrow, constriction. Right? So vasoconstriction is when the smooth muscle of an artery or a vein contracts to make the blood vessel narrower, right? And then if you do the opposite, you relax the smooth muscle, the blood vessel will expand until it has a larger diameter, and that's called vasodilation. Vasodilation. Right, so we learn more about vasoconstriction and vasodilation. We learn a lot more about that in Anatomy 130, but you don't have to know too much about that here, except that I probably will not ask you about vasoconstriction, vasodilation here. Okay, so the third layer of a blood vessel on the very outside is made of connective tissue, and it is simply there to give the blood vessel more strength. Okay, so notice that there's a difference between the thickness of the smooth muscle and the thickness of the layer of connective tissue between an artery versus a vein. So the vein is made of the same 3 layers. We have a layer of endothelium on the inside, surrounded by a layer of smooth muscle, which is surrounded by a layer of connective tissue. The layer of connective tissue and the layer of smooth muscles are much thinner in a vein. And the reason for that is because in an artery, the blood is just leaving the heart. And when the blood leaves the heart, it leaves the heart at high pressure. It has to leave the heart at high pressure in order to make it all the way around the body, which is a long distance. So when the blood is first leaving the heart, it leaves the heart under high pressure. After the blood has passed through capillaries and entered the veins, that high pressure is gone. So blood returns to the heart at low pressure. And so if you're returning, if the blood vessel is carrying blood at low pressure, there's no need to have such a thick layer of connective tissue and smooth muscle. So the layer on the inside, the very inner layer of a vein versus an artery is the same. It's a single layer of squamous cell epithelium that we refer to as endothelium. And then we have a layer of smooth muscle and a layer of connective tissue. Those layers of smooth muscle and connective tissue are thick when we're talking about an artery, and they are thin when we're talking about a vein. There's one other thing that you notice about the vein versus the artery. Notice that the vein has something here called a valve. Right? The blood passing through, let me tell you that a valve generally means something that ensures that a valve is something that makes sure that a liquid travels through a pipe or a vessel in one direction only. Right? So a valve is usually used to prevent backflow. Backflow means liquid flows in the wrong direction. Right? So we want the fluid in the vein, we want the blood in the veins to travel to the heart, but it's traveling at such low pressure and it's traveling so slowly that it may very well stop and then sometimes go in the wrong direction. So the purpose of the valve is that if the blood tries to go in the wrong direction, the valve will close. If the blood is traveling in the right direction, the valve will open. And so the valves that you find inside veins are really little more than a little tiny funnel. Imagine if you had a rubber funnel. And you poured water down that funnel, if the water traveled in the right direction, the funnel would be open. But if you had this very thin rubber funnel, if you tried to push water through it in the wrong direction, the rubber funnel would collapse rather. It would collapse under the pressure of the water trying to flow in the wrong direction. So that's exactly what the valves are in human veins. They are thin little rubber funnels that are made of squamous cell epithelium, and if the blood tries to travel in the wrong direction, they will close. If the blood is traveling in the right direction, they'll open. Okay, so the main differences between arteries and veins are that the arteries have a thicker layer of smooth muscle and a thicker layer of connective tissue, and they do not have valves. They're not necessary because the blood is traveling at very high pressure in the right direction. After the blood travels through the capillaries, the pressure is dissipated. The pressure disappears. So when the blood is traveling through a vein, you don't need a thick layer of smooth muscle or connective tissue, but you do need valves to make sure that this low pressure blood is traveling in the right direction to get back to the heart. Okay, about the capillaries. The capillaries are kind of like a webbing or a net shape because there's a lot of them. And their purpose is simply to surround the organ that they're delivering blood to, or to surround these, or to deliver oxygen to tissues. So because the network of capillaries in the body is so extensive, that's where the pressure gets dissipated. So the blood enters capillaries at relatively high pressure. Once it travels through, as it's traveling through the capillaries, the pressure disappears. And when the blood emerges on the other side of the capillary and enters a vein, like so, the pressure is gone. So the blood is traveling back to the heart at very low pressure. Okay, now there are different parts of the heart that we need to be aware of. Just pretend for a minute that the heart has a right side and a left side. It does, but each of those sides is divided into smaller chambers. But let's just say, let's look at the right versus the left side of the heart. Okay, the right side of the heart receives the oxygen depleted blood from the body and sends it to the lungs, right? The lungs are referred to, you know what a circuit is, an electrical circuit? A circuit means that you go around the whole thing and you come back to where you started from. You begin someplace, you flow around like electricity, and then you end up back at the beginning again. That's called a circuit. Right, so the pulmonary circuit refers to the flow of blood through the lungs and back to the heart. So blood flows from the right side of the heart. Oxygen depleted blood is delivered from the body into the right side of the heart, and the right side of the heart sends the blood through the lungs and then back to the heart again. When the blood returns to the heart after having traveled through the lungs, it returns to the left side of the heart. and then the blood is sent from the left side of the heart out to the body. So we call the journey through the lungs, we call that the pulmonary circuit, because we start at the heart and then we go through the lungs, and then at the end we end up back at the heart again, don't we? Right, so it's a circuit if you go through this course and then you end up right back where you started. So the blood travels from the heart through the pulmonary circuit, the lungs, back to the heart again. The only difference is that the blood starts in the right side of the heart, travels through the lungs, and then finishes in the left side of the heart. The left side of the heart then sends the blood out to the body, which is referred to as the systemic circuit. And the very first blood vessel where the blood leaves the heart traveling out to the body is a very large artery called the aorta. So there's a very large, powerful, strong, thick artery that connects, that delivers blood from the heart to the rest of the body, and that's called the aorta. So the aorta is the largest and the thickest artery in the body. The blood is just leaving the heart and traveling at very high pressure, so the aorta has to be very strong and have very thick walls in order to withstand the pressure of the blood as it leaves the heart. As the blood travels around the body, the pressure starts to drop lower and lower and lower, so other arteries and other parts of the body do not have to be so strong, but the aorta has to be very strong because it is carrying the blood as the blood leaves the heart for the first time at a very high pressure. Okay, so the systemic circuit refers to the body, the blood flowing around the body and then back to the heart, right? So systemic circuit. And the systemic circuit is actually divided in two. And so the systemic circuit, that means the aorta, at a certain point the aorta will divide in two and deliver some of the blood to one part of the body and the rest to the other. So the systemic circuit is divided into two halves. One half of the systemic circuit sends blood to the head and the arms. So it sends blood upward from the heart to the head and the arms. And the other part of the circuit sends blood to the lower body, the abdomen, the intestines, the kidneys, the liver, the leg muscles, and so on, all of that. Right, so the blood travels through the body and then back to the heart where it is returned. The oxygen depleted blood is returned to the heart, the right side of the heart, through two very large veins that are called the superior vena cava and the inferior vena cava. As you can guess from the word superior and inferior, the superior vena cava delivers oxygen depleted blood to the heart from the head and the arms. The inferior vena cava delivers the oxygen depleted blood from the lower body to the heart. Right, so the superior vena cava and the inferior vena cava are two very good examples of veins that deliver oxygen depleted blood back to the heart. Okay, so here's kind of a very simplified diagram. Here we have a bunch of capillaries that are meant to represent the systemic circuit of the heart. At the top, we have a bunch of capillaries that are lining the lungs. So this is the pulmonary circuit up here. Right, so after blood has been through the lower part of the body, it travels up through the inferior vena cava and gets delivered to the right side of the heart. Similarly, from the right side of the heart, the blood travels through the lungs and then back to the left side of the heart. And then from the left side of the heart, it travels out through the aorta and then through the systemic circuit one more time. Right, so the left and the right sides of the heart. The right side of the heart receives oxygen depleted blood from the body and sends it to the lungs. From the lungs, it travels back and ends up in the left side of the heart, and from the left side of the heart, the oxygen-enriched blood gets sent out to the systemic circuit, gets sent out to the body, and then it goes back to the right side of the heart, and so on. All right, so it's a little bit more complicated than that. That first diagram I showed you was a simplified version. Right, so the pulmonary circuit is actually divided in two because we have a left lung and a right lung. So you're seeing here the right lung. This person is standing in anatomical position, so we're talking, when I talk about the right lung, I mean the lung on their right. And then the pulmonary circuit also has a left side. So there's a right and a left pulmonary circuit. and we also know that we have a superior and an inferior systemic circuit. Right? So it's a little bit more complicated than that, but it's basically the same as that diagram I showed you on the last slide. All right, so here we have a better diagram, right? So we have we have one part of the systemic circuit delivering blood to all of the capillaries in the upper body. That includes the head and the arms and the chest. We have another part of the systemic circuit that delivers blood to the digestive organs as well as the leg muscles and things like that. And then we have a right half to the pulmonary circuit and a left half to the pulmonary circuit, because we're delivering blood to the right and the left lungs. And then all of that blood ends up back in the left side of the heart. All right, let's learn some of the names of the major blood vessels of the heart itself. Right, so the superior vena cava is a vein that delivers blood to the right side of the heart from the upper part of the body, the head and the arms. That's called the superior vena cava. There's another vein called the inferior vena cava that returns blood to the right side of the heart from the lower part of the body. All right, then we have the pulmonary arteries. We have a left and a right pulmonary artery that transport the blood from the right side of the heart to the left and right lung. And then we have the pulmonary veins, which deliver oxygen-enriched blood from the lungs back to the heart, specifically to the left side of the heart. And then we have the aorta that delivers blood to the body, and the aorta comes out of the left side of the heart. Now the heart itself needs blood, and so the arteries that deliver blood to the heart itself are called the coronary arteries. All right, so sometimes you might have heard that somebody had a coronary. When you say that somebody has a coronary, that's a kind of an abbreviation. That's a slang term meaning that they had a blockage of one of the coronary arteries. So there are several arteries that supply blood to the heart. Because the blood is responsible for pumping blood to the rest of the body, those arteries are very important, aren't they? Right? So if we can't supply blood to the heart, it's game over. Right? You would die if you couldn't have blood supplied to the heart. You wouldn't die if you lost the blood supply to a leg. The leg would die, but you could cut it off and you could still live. You wouldn't die if you had a blood supply was cut off to the liver. You could probably fix that before you died. But certainly if you cut off the supply of blood to the heart, you would be dead fairly quickly. And so that's what they mean when they say a coronary. Coronary is a slang term for a heart attack. A heart attack is usually caused because one of the blood vessels that supplies blood to the heart, one of the coronary arteries, has been blocked, and it has cut off the supply of blood to the heart. So that is sometimes referred to as a coronary. A coronary means a heart attack. A heart attack is usually caused by one of the coronary arteries being blocked, so that the supply of blood to the heart is blocked. All right, so the blood that goes to the heart has to come back to the heart again. So the coronary arteries deliver fresh oxygen-enriched blood to the heart muscle, to the myocardium itself, and then the cardiac veins deliver the oxygen-depleted blood from the heart back to the heart, right? So the oxygen-depleted blood is delivered from the cardiac veins back to the right side of the heart, where it gets pumped through the lungs. All right, now you remember I told you that there were some notable exceptions to the idea that arteries deliver oxygen-enriched blood to the body. Arteries usually carry oxygen-enriched blood, but there are some notable exceptions, and one of them is the most notable exception is the pulmonary artery. The pulmonary arteries carry oxygen-depleted blood from the right side of the heart to the lungs, and so they are called arteries, but they are carrying oxygen-depleted blood. That's the most oxygen-depleted. The blood traveling through the pulmonary artery has the absolute lowest oxygen content of any blood vessel in the body, right? Because it's about to travel back to the lungs and get recharged with fresh oxygen. Okay, and then here's an example. The pulmonary veins are an example of a vein that carries oxygen-enriched blood, because the veins are carrying blood from back to the heart from the lungs, but that blood is very oxygen-enriched, isn't it? Right. So there are two notable exceptions. It is generally true that arteries carry oxygen-enriched blood and veins carry oxygen-depleted blood, but there are two very strong exceptions to that. The pulmonary arteries carry oxygen-depleted blood to the lungs from the heart to the lungs, and the pulmonary veins carry oxygen-enriched blood from the lungs back to the heart. Okay, so there's our circuit again. There are the coronary arteries in red and the cardiac veins in blue. Generally, when blood is carrying oxygen, it's red in color, and when it's oxygen depleted, it's bluish in color. It's a darker shade. It's more like a darker shade of purple, but sometimes it can actually be blue. And you may wonder, if you cut open, you can see veins on your wrists and different parts of your body, and if you poke one of those veins, the blood that comes out will be red, not blue. If you have very fair skin like I do, for instance, I can actually see on the veins on the back of my hand that they're blue in color, right? So I can see these I have very fair skin, so you can actually see these blue lines on the back of my hands. Those are the veins, and if you poke one of those veins, like with an IV needle in a hospital, you would see that the blood comes out red. And the reason for that is as soon as you poke open the vein and the blood comes out into the atmosphere, there's oxygen in the air, so that blue blood automatically turns red because suddenly the hemoglobin will pick up the oxygen, right? So you never see blue blood outside of a blood vessel, but you can see blue blood in veins, provided that it's not exposed to oxygen. Okay, so here we have, so generally on anatomy diagrams, the blue blood vessels are meant to be veins that are carrying oxygen-depleted blood, and the red lines are meant to be arteries that are carrying oxygen-enriched blood, because oxygen-enriched blood is red in color and oxygen-depleted blood is blue in color. Okay, so there's our circuit. Okay, now I talked about the right and the left sides of the heart. The right and the left sides of the heart are each divided into two additional parts. There's an upper part that is called an atrium. Now here we go with the Latin words again. You know what an atrium of a building is? You know, if you've been in Columbia College, you know that area where you enter the building and then climb up the stairs, you walk up the stairs, is called the atrium. Right? So the atrium, an atrium is a Latin word meaning the room that you enter before you enter the main part of a house, right? So you go into the atrium of the house. If you're in ancient Rome, you go into the atrium of the house and you take off your sandals and you take off your cloak, and then you get cleaned up a little bit and compose yourself before you go into the main part of the house. Right. Okay, so an atrium in the heart, if you look at the heart, there are two chambers at the top of, there's a chamber at the top of the right side of the heart, that's called the right atrium. There's another chamber at the top of the left side of the heart, that's the left atrium. And then from the atria is plural, of atrium is single, atria is plural. From the atrium, the blood will go downwards through a valve into another chamber that is called the ventricle. Right. The atria are surrounded by a thin layer of myocardium. That means that they have a fairly weak, they can only generate fairly weak pressure. And basically the blood goes into the atrium and waits there until it gets permission to go into the main room of the heart, which is the ventricle. Right, so an atrium is a room where you enter the house and you wait until you get tidied up, and then you get permission to go into the main rooms. Right, so the atrium, in the case of blood, the blood goes into the atrium and then waits before it has permission to go into the ventricle. The ventricles are surrounded, there are two ventricles, a left and a right ventricle. The ventricles are surrounded by a very thick layer of myocardium, and they provide most of the pumping action for the heart. So the atria, their only job really is to squeeze the blood into the ventricle, and then once inside the ventricle, the ventricles will contract and pump the blood through the heart or out of the heart with a tremendous force, because there is a thick layer of myocardium surrounding each of the two ventricles. And the heart has two valves within the heart, and those valves prevent backflow when the heart beats. So normally we would have blood flowing from the right atrium into the right ventricle, and from the right ventricle it goes into the lungs. It goes towards the lungs. Okay, but what we don't want is for the blood to go from the right ventricle back into the right atrium. So there is a valve that separates the right atrium and the right ventricle. If the blood pushes in the wrong direction, trying to get back up into the atrium, that valve will slam shut. And so the blood only has one direction to go, and that's the other direction towards the heart. Right? So that's the purpose of the valves. Because we have two sides to the heart, we have a left and a right atrium, and we have a left and a right ventricle. There is a valve located between each of the ventricles and between each of the atria and its respective ventricle, right? So there is a valve that separates the right atrium and the right ventricle, and there's another valve that separates the left atrium and the left ventricle. Because these two valves are separating atria from ventricles, we call them atrioventricular valves. Right? So the heart contains two atrioventricular valves. There's a right atrioventricular valve, and there's a left atrioventricular valve. The right atrioventricular valve has a special name, and so does the left atrioventricular valve, and we'll talk about those. The right atrioventricular valve is actually called the tricuspid valve, and the left atrioventricular valve is called the mitral valve. All right, let's take a closer look. So here we have the superior vena cava, and here we have the inferior vena cava. The superior vena cava is delivering blood to the right atrium, and the inferior vena cava is delivering oxygen-depleted blood to the right the right atrium as well, the inferior vena cava. So the oxygen-depleted blood from the body ends up here in the right atrium. The right atrium will squeeze that blood into the right ventricle in this direction. Notice that the two ventricles are surrounded by a very thick layer of myocardium that is around the bottom of the heart. The bottom of the heart is actually called the apex of the heart. And there's a thick layer of myocardium separating the middle of separating the two ventricles, and that layer is called the septum. Septum is a Latin word meaning wall. Right, so the septum, the middle part that separates the two ventricles, is called the septum. Okay, so in fact, because it's separating the two ventricles, it's specifically called the ventricular septum. The ventricular septum is a thick layer of myocardium that separates the left ventricle from the right ventricle. There is a thinner layer of myocardium that's called the atrial septum that separates the left atrium from the right atrium. Right, so those are the septi. Okay, so we've taken the blood, sent it through the superior vena cava and the inferior vena cava, where it ends up in the right atrium. All of this oxygen-depleted blood ends up in the right atrium. The right atrium squeezes the blood through this first atrioventricular valve into the right ventricle. As I said earlier, the right atrioventricular valve is referred to as the tricuspid valve. Now, the word tri means three, and the word cuspid means leaflet, right? So if you ever see this valve, you'll see that it has three little triangular leaflets that are on the ventricular side, and those three little leaflets will slam shut if the blood tries to go in the wrong direction from the ventricle back into the atrium. Right, so the tricuspid valve is named the tricuspid valve because it's made of three cusps. It's made of three triangular shaped flaps of connective tissue called cusps. All right, so now we have the blood squeezed from the atria into the right ventricle. And then when the ventricle contracts, that myocardium squeezes, it will push the blood with great force through this blood vessel, which is called the pulmonary trunk. And as it goes through the pulmonary trunk towards the lungs, the pulmonary trunk bifurcates. You remember that word, bifurcates? It divides into two pulmonary arteries. that are traveling towards the lungs. Okay, so the blood travels from the right ventricle through another valve that prevents blood flow in the wrong direction, prevents backflow, and the blood flows from the right ventricle into the pulmonary trunk. The pulmonary trunk bifurcates into a left and a right pulmonary artery. Okay, what's that other valve called? So this other valve that prevents backflow from the lungs back into the right ventricle is called it's called the pulmonary semilunar valve. Okay, so there are two valves that are in the body. The word semilunar means half moon. You've seen a crescent moon, right? So if you look at those valves, they look sort of like a half moon in shape if you're looking down at them. Not really, but somebody thought they did, so they gave them those names, right? So the pulmonary semilunar valve is the valve that prevents backflow from the lungs back into the right ventricle. Right. So then the blood goes through the lungs, and then it comes back into the left atrium, right? And then the blood travels from the left atrium. It's squeezed through a valve called the mitral valve into the left ventricle. So the blood goes from the lungs back into the left atrium. From the left atrium, it's squeezed through the mitral valve into the left ventricle. And then when the left ventricle contracts, it pushes the blood with great force through another valve, another semilunar valve, which is called the aortic semilunar valve, or the aortic valve. And then it travels into this blood vessel, the aorta. So the aorta is very thick and strong because the blood is coming out of the heart with great force. And so it's the strongest artery in the body. So the strongest artery in the body is the aorta. Now notice that the aorta has these three little things coming off the top. Those three little arteries are headed towards the head and the arms, right? So we'll talk about those later. And then the rest of the blood that didn't go up to the head or the arms is heading downwards through the aorta as it goes downwards to the lower part of the body. Okay, so let's look at the valves one more time. The tricuspid valve is located between the right atrium and the right ventricle. It prevents backflow to the right atrium from the right ventricle. The pulmonary semilunar valve prevents backflow from the lungs back into the right ventricle. The mitral valve is located between the left atrium and the left ventricle, and it prevents blood from flowing backwards from the left ventricle into the left atrium. And the aortic semilunar valve prevents backflow of blood from the body back into the left ventricle. Okay, so there are the four valves: the tricuspid valve, the pulmonary semilunar valve, the mitral valve, and the aortic semilunar valve. I will not ask you about this in this course, but the mitral valve has another name called the bicuspid valve. Okay, why do you think if we call the tricuspid valve, what do you think the shape of the bicuspid valve is? If you guess the fact that the mitral valve is composed of two cusps, has two little triangular shaped leaflets, you would be correct. So we have a tricuspid valve and a bicuspid valve. The bicuspid valve, I like to call it the mitral valve because it's too easy to confuse the word bicuspid with tricuspid. But in Anatomy 130, Biology 130, we do get you to memorize the word bicuspid, and to note we require that you know that the bicuspid is another name for the mitral valve. All right, let's take a little imaginary journey, supposing that you were one or two blood cells. Suppose that you were one or two oxygen-depleted blood cells, and you're going to go through the heart and the pulmonary circuit and back out to the body again. Let's look at all the structures that you would encounter as you're going on this journey. All right, so we have all the structures that we're going to come in contact with on the right-hand side of the slide, and on the left-hand side we have a picture of the heart. Starting off, oxygen-depleted blood cells, which are blue in color, are sent using the superior vena cava and the inferior vena cava into the right atrium. Once they're in the right atrium, They're going to go through the right atrium into the right ventricle. As they do this, they go through the tricuspid valve and they end up in the right ventricle. Next, they're going to go out through the pulmonary trunk to the lungs. So they have traveled through the pulmonary semilunar valve, and they're now in the pulmonary trunk, and they're just about to head out either through the left or the right pulmonary artery to the lungs. After they come back from the lungs, they travel through the left and right pulmonary veins, and they're going to end up in the left atrium. Now they're in the left atrium. They're going to travel through the mitral valve to the left ventricle. They've traveled through the mitral valve. They're in the left ventricle. Next, they're going to travel through the aortic semilunar valve to the aorta and then out to the body. Right, so they have just traveled through the aortic semilunar valve. They're now in the aorta, and from there they go out to the rest of the body. Okay, so that's the journey of blood through the heart. Right, so one more time, here's a diagram of the heart. Please study it, because I could show you a diagram and say label all these parts. All right, here's a little exercise. Which of these valves and blood vessels are in contact with oxygenated blood and which are in contact with deoxygenated blood? Right, so I'll do some of it, but not all of it. So you should try this exercise on your own. You can pause the tape and then see if you can remember whether it is oxygen enriched blood or oxygen depleted blood. Oxygen enriched blood is sometimes called oxygenated blood. So which of these valves are in contact with oxygenated blood? Let's ask that. Okay, so you can pause the recording and then start when you think you have the answers. All right, let's go through the answers. Right, so the superior vena cava, is it in contact with oxygenated blood? No, it's not, because the blood is oxygen depleted as it travels through the superior vena cava, so not that. What about the tricuspid valve? Oh, it's still depleted. What about the pulmonary semilunar valve? Still depleted. Okay, pulmonary artery. Okay, so it's heading from the heart to the lung, still depleted. Pulmonary vein, yes. So the pulmonary vein is carrying oxygenated blood from the lungs back to the heart. As the blood travels between the left atria and left ventricle, it travels through the mitral valve, so the mitral valve is in contact with oxygenated blood. As the blood passes from the heart to the aorta, it travels through the aortic semilunar valve and it's oxygenated, and through the aorta, oxygenated. Okay, so this is the color code. The blue means oxygen depleted, the red means oxygenated or oxygen enriched. That was easy because I put them in the right order. Why don't we scramble them into the incorrect order and see if you can get it right? Okay, you stop the recording and then I will give you the answers afterwards. Indicate whether each of these structures is in contact with oxygenated or oxygen depleted blood. All right, I'm going to give the answer now. So the blue structures are in contact with oxygen depleted blood, the red ones with oxygen enriched. Okay, so that could be something I ask you to do on a test. All right, now congenital heart defects, as I said, I think it's useful to learn about some problems that you have with each of these organ systems, pathology, because if we study how and why these organs have problems, and we study what happens when they're working improperly, that helps you to understand how they work properly. So a congenital heart defect, it has the word gene in it. Gene means the beginning. It's a Greek word meaning from the beginning, right? So it has the word gene, but that does not mean that these are necessarily genetic defects. Some of these are simply, some of these defects that I'm going to show you are not genetic. Genetic means that it's passed from one generation to another. Most of these defects are not. They're not. They just happen by accident. But you know, somebody who has a congenital heart defect, they have a problem with the heart from the beginning, but it's not something that they would pass on to their children necessarily. There are genetic defects, and we'll talk about those when we get to the reproductive system, that are defects and problems that are carried in the genes and can be passed from one generation to the next. But the congenital heart defects, the ones that I'm about to talk about here, are not genetic, meaning that if you have a congenital defect with your heart, you have a problem with your heart, you will not necessarily pass it on to your children. Okay, now a very common congenital heart defect is an atrial septal defect, or an ASD. Defect means, you know what the word septal means? It refers to the septum. And if we talk about if we use the words atrial septal, it means we're talking about the septum that divides the right atrium from the left atrium. A defect is a fancy word in this case. In this case, defect means a hole. Right? So there's a hole between, you are born with a hole in your heart that connects the left and the right atrium. That's called an atrial septal defect, which is sometimes abbreviated ASD. You do need to know that. And then there's another septum that separates the right and the left ventricles, which is much thicker. And that's called, if you have a hole in the ventricular septum, it is called a ventricular septal defect, or a VSD, right? Ventricular septal defect. Okay, now most of the pressure that is generated by the heart is generated by the ventricles, right? So remember that the atria, their only job is to squeeze a little bit of blood into the ventricle, and then the ventricle provides a tremendous pumping action that shoots the blood either to the lungs or out into the body. So most of the pumping action is provided by the ventricles. What that means is that if you have an atrial septal defect, you have trouble building up pressure in the atria, but who cares? Because the atria don't need to generate much pressure anyway. But if you have a hole between the left and the right ventricles, the pressure is kind of released by the hole. That's a serious problem. So that means that atrial septal defects tend to be less serious than ventricular septal defects. Ventricular septal defects tend to be a much bigger problem than atrial septal defects. All right, so let me describe to you three different types of, first of all, let me ask, what is shown in this picture? In this picture, are we looking at an ASD or a VSD? You tell me. Right? And if you answered that we're looking here at an atrial septal defect, you would be right. All right, so there are examples of both that people are frequently born with. Okay, and so they have to be fixed. Okay, usually they have to be fixed. Okay, so I'm going to tell you about three different septal defects, congenital defects of the heart. The first one is called patent foramen ovale, PFO. Patent foramen ovale, and I will show you how to spell this in a minute on the next slide. Patent foramen ovale is a hole between the two atria. So a patent foramen ovale is a hole in the atrial septum. So a patent foramen ovale is an example of an atrial septal defect. And because I said that, why are you born with a hole between the atria? Well, when you're a developing fetus, the lungs have not fully developed, and so the circuit, the pulmonary circuit, is bypassed in a developing baby that has not been born yet. So when all of us were developing in our mother's womb, we used to have a hole. We had a deliberate hole between the two atria, because the blood traveling through a developing fetus's body is meant to bypass the baby's lungs. Right? So it's not meant to go through the lungs because the lungs are still developing. That hole that's intended to bypass the lungs is meant to close before we're born. Sometimes it fails to close before we're born, and you end up with something called patent foramen ovale. The word patent is a Latin word for meaning a hole that is open rather than closed, right? A patent foramen ovale, the hole that fails to close, is referred to as the foramen ovale. Ovale is a Latin word, just means oval shaped, right? So we all ordinarily have an oval shaped hole that connects our left and right atria before we're born. That hole is supposed to close. The hole itself is called the foramen ovale. If it fails to close by the time we're born with that hole open, so it's called a patent foramen ovale. Okay, another septal defect is called a patent ductus arteriosus. I will show you how to spell that as well. Okay, so again, because the bloodstream in a developing fetus is meant to bypass the lungs, it's meant to go around the lungs instead of through it until the lungs are finished developing, we have a hole that connects our pulmonary trunk to the aorta. Right, so we have while we're developing in our mother's womb, we have two holes in our heart that are supposed to be there. One of them connects the aorta to the pulmonary trunk, and one of them connects the two atria. Those two holes are meant to allow the blood to go around the baby's developing lungs instead of through the developing lungs, right? That's the idea. And those two holes are supposed to close. The hole that's connecting the two atria is called the Foramen Ovale. The hole that's connecting the atria, the hole that is connecting the aorta and the pulmonary trunk is called the ductus arteriosus. So the Foramen Ovale and the ductus arteriosus are supposed to close before we're born. If either one of them fails to close, you either have patent Foramen Ovale or patent ductus arteriosus. Okay, so patent Foramen Ovale, a hole between the two atria; patent ductus arteriosus, a hole connecting the pulmonary trunk to the aorta. If you have a really bad congenital heart defect, it's called Tetralogy of Flow. Again, I'll show you how to spell that later. Tetralogy of Flow is where you have both a ventricular septal defect and a reduced diameter of your pulmonary trunk. That means that the pulmonary trunk that takes blood from the heart to the lungs is narrower than it should be. If it's narrower than it should be, you can't pump enough blood around the body. And furthermore, you can't develop enough pressure in the heart anyway, because you have a hole connecting the left and right ventricles, not the atria this time, but the ventricles. Right, so if you have a reduced diameter for your pulmonary trunk plus a hole connecting the left and right ventricles, you have a very serious condition called Tetralogy of Flow, and so you have to have both of those. Problems fixed by doing surgery on the heart, and doing surgery on a baby's heart is very tricky, very dangerous. So in fact, the baby's blood can't build up enough pressure to move around the body, and the pulmonary trunk is narrow, so the blood has trouble getting oxygenated. And so when a baby is born with Tetralogy of Flow, it comes out looking a little bit blue because the oxygen is having trouble, the blood is having trouble getting oxygen. And so that is sometimes called blue baby syndrome. And we're going to learn about two different blue baby syndromes in this course. Tetralogy of Flow is an example of one blue baby syndrome, and the other one is a result of having the wrong blood type, which we'll learn about in a minute. Okay, so let's look at these congenital heart defects in greater detail. Okay, so as I said, a patent Foramen Ovale is a hole between the left and right atria. It's usually not terribly serious. So in fact, about 20% of the people in the world have a patent Foramen Ovale, and they don't even realize it because it's not enough of a problem that they would ever have gone to a doctor and had it diagnosed. So 2 out of every 10 people has a patent Foramen Ovale, and the only symptom that they had was that they tired easily, right? So one of the symptoms of patent Foramen Ovale is that you fatigue easily, you get tired easily, and you probably weren't very good at sports because you tired easily, and that's you never thought twice about it. You just thought, well, I'm not going to be a soccer player, I'll do something else for a living. And so you were one of the poorest athletes at school, and you didn't think that there might be a reason why, but it could have been because of patent Foramen Ovale. Okay, patent ductus arteriosus is where you have the artery connected to the pulmonary trunk, which is very serious indeed. It's extremely serious. So that has to be fixed by surgery. Tetralogy of Flow, again, is a very serious problem where you have a reduced diameter to the pulmonary artery or the pulmonary trunk, plus a hole connecting the two septum of ventricular septal defect. And so babies that are born with Tetralogy of Flow usually come out looking a little bit blue because they're having trouble getting oxygen into their blood. All right, let's spend a few minutes talking about heartbeats. Let's spend a few minutes talking about how the heart beats. Okay. Overall blood pressure, when the heart beats and sends blood to the lungs or to the body, it generates high pressure. So when the heart beats, it squeezes the blood out at high pressure. And so the blood traveling through the body is temporarily at high pressure, and we call that systole. right? So when the heart squeezes the blood out and generates high pressure, we call that systole. On the other hand, when the heart relaxes to let blood in, there's a temporary low pressure to the blood, which we call diastole, right? So the pulse is the rhythmic switching between systole, diastole, beat, relax, systole, diastole. That is what generates what we call a pulse. Okay, so make sure that you know that systole is squeezing, pumping blood out, high blood pressure. Diastole is relaxing, low blood pressure. And the rhythmic beat between high and low blood pressure, high, low, is systole, diastole. Right? So there is a way to measure the pressure of the blood when it is at systole versus diastole. You can measure the blood in both cases, the blood pressure in both cases, and I will show you how to do that. All right, so when you measure a pulse, this, by the way, is you push your fingers against the radial artery. So it's called the radial artery. It's normally used to take the pulse. It's called the radial artery because the artery, it runs just along the top of the radius, the radial bone in the arm. So you put your two fingers there and you can feel your pulse, right? So you're feeling your radial artery, right? So that's what you're feeling. Blood is, every time you feel a beat, it's because the blood is squeezing blood out and generating a temporary transient high blood pressure called systole. All right, now the heart beats in 3 distinct beats. If you've seen a heart beat, it doesn't look like it's just squeezing, relaxing. It looks like it's wobbling. It looks like it's wobbling about. And that's because the different parts of the heart, in fact, the atria are squeezing at a different time than the ventricles are squeezing, right? So the atria squeeze first, and then the ventricles squeeze, and then the whole heart relaxes, right? So remember, squeezing we call systole, relaxing we call diastole. Okay, so the heartbeat occurs in 3 distinct steps. The first step is called cardiac diastole. Cardiac diastole is where the whole heart relaxes to let blood in. Then we have atrial systole, ventricular diastole. So that means during the second stage, the atria squeeze the blood into the ventricles, right? So that's the first part. The next part, the third step, is atrial diastole, ventricular systole. That means that the atria relax while the ventricles squeeze and push the blood out, right? So first, cardiac diastole, the entire heart relaxes. Next, the atria squeeze, and next, the ventricles squeeze, right? So relax, squeeze the atria, squeeze the ventricles. 3 steps, and that gives you that kind of do-do that you hear if you listen to the heart with a stethoscope. All right, so cardiac diastole, the entire heart relaxes in order to let blood in. Atrial systole, ventricular diastole, the ventricles are relaxed while the atria are squeezing. And then the third step is atrial diastole, ventricular systole, the ventricles are squeezing while the atria are relaxing. All right, you can actually measure the heartbeat, and frequently do, using a machine called an electrocardiogram. So electro refers to electricity, cardio refers to the heart, and the word gram usually means picture or graph, right? So electrocardiogram, abbreviated ECG, is where you put electrodes. Electrodes are little electrical sensors. You put them on either side of the chest, and because when a muscle contracts, it generates a little bit of electricity. Electricity is not necessary, but it's just a side effect. It's something that happens incidentally. So you can measure the electricity that's generated as the heart muscles are contracting, and you can actually plot a graph of that called an ECG. There's an older convention from the 60s where they used to call it an EKG, because the German word, it was perfected in Germany, and so the German word for cardio is spelled with a K instead of a C. So they used to call, in the old days, they used to call it an EKG. If you look at some of the old medical shows, medical soap operas that used to be on television from the 60s, they would always call it an EKG. That term is considered to be obsolete. The proper terminology is an ECG, for electrocardiogram, cardio spelled with a C. Okay, so ECG is an electrocardiogram. Okay, so you put electrodes on the chest, which are capable of measuring the electrical signals that are generated when the heart beats, and then you plot it out onto a graph. And you see sometimes instead of plotting it onto a graph, you display it on a little TV called an oscilloscope that gives you a little green line like this. So that's an oscilloscope version of the ECG. Right? So you see a sharp peak in the middle and then some smaller peaks. We're about to learn what those mean. We're about to learn what those little parts of the ECG wave, mean. Okay, so they are actually labeled P, Q, R, S, and T, right? So P is the electrical signal that's generated when the atria contract, right? So the P part of the ECG wave is the atrial systole. Okay, then the signal moves to another part of the heart, which I'll show you in a minute. I'll show you a diagram. The signal moves to the bottom of the heart, which is called the apex. And then we have the ventricular contraction, which is quite dramatic, a big hard push when the ventricles contract. That is called the QRS part of the ECG wave. And then when the entire heart relaxes, cardiac systole, that is called the T part of the wave. All right, so that means the P part, P, means atrial systole. The atria are squeezing the blood into the ventricles. The QRS part of the ECG wave, the QRS part is where the ventricle contracts and creates systole. Right? And then the T part is where the whole heart relaxes, cardiac diastole. So the whole heart relaxes to let new blood in. Right, so P, Q, R, S, T. You are expected to know which part of the ECG wave represents which part of the heartbeat. Okay, this is just showing you what's happening with the electricity. The electricity starts, the heartbeat originates in a structure that's called the sinoatrial node, which is commonly called the pacemaker. Right, so that's where the electrical signal that causes the heart to beat begins. Right, and that is represented by this part of the ECG wave. All right, the signal that is then transferred to another spot called the Atrioventricular Node. That's actually this part of the ECG wave, but I don't expect you to remember that. And then the signal is transmitted down the ventricular septum to the base of the heart, which in this case is called the apex, the heart apex. That's what this is. That's what's represented by that. And then when you have this huge depolarization, this huge squeezing, beating of the ventricles, that's the QRS part of the ECG wave. So we have P, QRS, and then T, which is not shown on this diagram. Okay, so that's what it means. All right, so if your heart is beating at the regular rhythm, as you know, normal, that is called normal sinus rhythm. Normal sinus rhythm, because the different parts, some of those different parts of the heart are referred to as sinuses. Don't worry about it, the term, but you are expected to know that the term normal sinus rhythm refers to a normal heartbeat. A cardiac arrhythmia is an irregular heartbeat. Okay, so here we have normal sinus rhythm on the left. And notice how irregular this is. Notice that the squeezing of the ventricles, which is the large sharp peaks, is very erratic. So in normal sinus rhythm, the time taken between QRS, the time taken between the ventricular systoles, is a constant amount of time. Notice that the amount of time between ventricular systole in the cardiac arrhythmia is irregular. Sometimes there's a long period of time between beats, sometimes there's a short period, right? So we call that a cardiac arrhythmia. All right, so normal sinus rhythm on the left and cardiac arrhythmia on the right, right? So in this case, when you have an irregular heartbeat, if either the atria or the ventricles are kind of, instead of beating the way they should, they're beating in a disorganized way, that is called fibrillation. Fibrillation. Fibrillation means irregular, kind of sporadic beating, right? Disorganized beats. Now, as it turns out, the disorganized beating can either be something that the atria are doing or that the ventricles are doing. If you have irregular beating of the atria, that's called atrial fibrillation, isn't it? Right? What do you think you would call it if the ventricles are squeezing at an irregular beat? Right? If you guessed, you would call that ventricular fibrillation. You're correct. Right? So sometimes atrial fibrillation is abbreviated as AFib. So on those medical television shows, you hear people talk about AFib or atrial fibrillation. And if it's ventricular fibrillation, they abbreviate it by saying VFib. As you can guess, VFib, ventricular fibrillation, is much more serious than atrial fibrillation because the ventricles are much more important at squeezing the blood, sending the blood, generating the pressure to send the blood to the lungs versus the heart. Right, so atrial fibrillation is much less serious than ventricular fibrillation. So ventricular fibrillation is much more serious than atrial fibrillation. In fact, generally, atrial fibrillation is sometimes caused by things that are as simple as a lack of potassium in your diet, right? So you need to eat more bananas to cure your atrial fibrillation, but atrial fibrillation is easily fixed with drugs. Ventricular fibrillation is rare, and if you have ventricular fibrillation, you're in danger of having a heart attack. So you generally have to do something about that very fast. If you're a doctor or a nurse or a paramedic and you have your patient is going into ventricular fibrillation, you have to use a machine called the defibrillator, which gives an electrical shock to the heart. All right, so cardiac arrhythmias are irregular heartbeats. Some other terms that you need to know: tachycardia. Tachycardia means a fast heartbeat, generally too fast, and too fast is generally means more than 200 beats per minute, right? So you know that in order to have a normal heartbeat, I just told you that tachycardia is an abnormally fast heartbeat, which is more than 200 beats per minute, right? So that means to have a normal heartbeat, you have to have a heartbeat that's less than 200, right? You can figure that part out now. So 200 beats per minute or more is called tachycardia, an abnormally fast heartbeat. You usually have that when you're excited or afraid, or you've been scared, or you've been running hard and you've run yourself ragged. You may have tachycardia, but it usually goes away as soon as all of the cells in your body get the proper amount of oxygen. Right. Then bradycardia is an irregular slow heartbeat. Now that's usually slower than 40 or 30 beats per minute, but there's a caveat to that, and the caveat is that athletes normally have a very slow resting heartbeat. So if you're a marathon runner, it's not unusual to have a resting heart rate of 40 beats per minute, so that's not an example of bradycardia. Fibrillation means an irregular heartbeat, and then you can have atrial fibrillation versus ventricular fibrillation. Ventricular fibrillation is much more serious, and you would have to fix that as an emergency measure with a machine called a defibrillator. Right? So a defibrillation is where you deliver an electrical shock to the heart, and then you just hope that it returns to normal sinus rhythm. Right? You've seen this on the medical shows and things. This is called a defibrillator. That's when the doctor puts the two paddles on either side of the chest and says clear, and then they deliver a shock that usually makes the patient convulse. That's the equivalent of rebooting a computer, right? If your computer crashes, you don't know what went wrong with it. You just hope that if you unplug it and plug it back in and turn it back on, that it will start functioning normally. Right? That's exactly what defibrillation is, except that you're rebooting the heart. Now, you have to be careful about this, because if you don't know what you're doing, if somebody's not having a heart attack, if you'd use the defibrillator on them, you may cause them to have a heart attack, right? That's why when the doctor or the paramedic puts the paddles on somebody's chest, they say clear. That means everybody else who's touching that patient has to take their hands away, and then they send the shock through. If somebody is still touching that patient, the shock will go into them, the electrical current will go into them, and you will cause them to have a heart attack. Right? So that's why in the old days they only let paramedics operate the defibrillator, because you had to be an expert to know that somebody was really having a heart attack. If they just had indigestion or they had some other pain that was not a heart attack, and then you use the defibrillator on them, you could actually cause them to have a heart attack. And at some point in history, I guess we decided that it was better to have, you would save more lives if you allowed amateurs, untrained people to use the defibrillator. So now in most gymnasiums around North America, in the gymnasium or in other places where people might have a heart attack, you will find that there's a first aid kit, and the first aid kit includes a defibrillator, and there's a little video that plays and tells you how to use, somebody who's never even seen one before is being told how to use the defibrillator. So I think, probably that may lead to some heart attacks, but probably they've made the calculation that it's worth it because you save a lot more people by having amateurs have access to the defibrillator than you would kill by accident. They might have also, I'm not an expert in this field, so they might have also made some improvements in defibrillators so that they've adjusted the current and the voltage so that it's less likely that you'd kill somebody by accident if you use the defibrillator on somebody who is not having a heart attack. All right, let's talk about the characteristics that all blood vessels, or both veins and arteries, have in common. Okay, so when I said all blood vessels, I actually made a mistake. What I'm about to say here applies to the arteries and to the veins, but not to the capillaries. Okay, now, first of all, the lumen, the hollow space inside a blood vessel, is referred to as the lumen. In fact, in all biological systems, whenever you have the inner hollow part of an organ or an organelle, it is referred to as the lumen. So, for instance, at the beginning of the course, we talked about the endoplasmic reticulum. So the space inside the endoplasmic reticulum, which is one of the cell organelles, the space inside the endoplasmic reticulum is called the lumen of the endoplasmic reticulum. Similarly, the inside of the intestine, the hollow part inside the intestines is called the lumen of the intestines, and the hollow part inside a blood vessel is also called the lumen. All right, now I mentioned the fact that there's a layer, so you have the lumen, so we're going from the inside out. You have the lumen at the center of a blood vessel. And then the first layer that lines the inside of the blood vessel is a thin layer of squamous cell epithelium. We call that, as I said before, when we find a single layer of squamous cell epithelium inside a blood vessel, we give it a special name. We call it endothelium. Right? That's what endothelium is. And there's yet another name that you have to remember for the squamous cell epithelium that lines the inside of a blood vessel. It is also referred to as the tunica intima. Tunica intima is a Latin word meaning undershirt. Right? So you remember the Romans wore tunics, and so if you had a little tunic that you wore close to your body, the word intima means close to you. Right? So the tunica intima means undershirt. Right, so the tunica intima is the inner layer of a blood vessel, which is made of squamous cell epithelium, which we then relabel, we rename endothelium, not epithelium, endothelium. And of course the word endo means inside, the word epi means on top of or above, right? So this is squamous cell epithelium, but because it's on the inside, we call it endothelium. Okay, then on the very outside we have the layer of connective tissue, which is also known as tunica externa. That's the Latin word for overcoat. And then the middle layer is smooth muscle, and it is also known by the name tunica media, middle tunic. Right, tunica media. So the middle layer, the tunica media, is made of a layer of smooth muscle, which is responsible for vasoconstriction, that is the narrowing of blood vessels, and also for vasodilation, that's widening of blood vessels. Generally, when you constrict the blood vessels, when you do vasoconstriction, the blood vessels get narrower. That causes the pressure of the blood to go up. Right? And then when you widen the blood vessels, the pressure of the blood goes down for exactly the same reason that when if you put a nozzle on a hose, you take a garden hose and you're going to use the garden hose to spray water across your yard or something like that. You screw something called a nozzle onto the garden hose, and then you can twist the nozzle so that it opens wider or narrower. Right? So you remember that if you do that, when you twist the nozzle so that the opening is narrower, the water shoots out of the nozzle with greater velocity. The water shoots out of the nozzle more quickly. And then when you open the nozzle up so it's wider, the pressure of the water coming out becomes less and less. Right? So exactly the same thing is true if you narrow or widen the blood vessels. Okay, so vasoconstriction is usually accompanied by higher blood pressure, and vasodilation is usually accompanied by lower blood pressure. Okay, so those are the layers of a regular vein or a regular artery. As I said earlier, the capillaries are made of just the tunica intima, or the endothelium. Right? So there is no layer of smooth muscle or connective tissue for the capillaries, and the reason for that is because the capillaries have to be thin enough that oxygen and carbon dioxide can cross, because the whole purpose of capillaries is to deliver oxygen to the organs and the tissues and to take away the carbon dioxide. So if we had a thick layer of smooth muscle and connective tissue, those gases would not be able to diffuse across that layer. So we need a thin layer of endothelium only in order to allow for gases to diffuse across. Okay, so here's another diagram. So here you can see the tunica intima, the lumen on the inside, right? The lumen on the inside, the tunica intima, the tunica media, smooth muscle, and the tunica externa, the connective tissue. This is a vein on the right-hand side. You can see the valve inside the vein. That, of course, is there because the blood pressure is quite low in veins, and therefore there's a danger that the blood will flow in the wrong direction. Okay, so here's another diagram. So here we have an artery. You can see the three layers. Here's a vein. You can see the same three layers except that they're thinner rather. You can see the valve inside the vein. And then here in the capillaries, the capillaries are very small and they have very thin walls to allow gas diffusion. Okay, once again, characteristics of arteries. Arteries carry blood away from the heart at high pressure. As a result of the high pressure, they have thick walls of connective tissue and smooth muscle. You can feel a pulse inside an artery. You cannot feel a pulse inside a vein. Veins, if you cut an artery, the blood will pump out, squirt, 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 because it's coming out with a pulse. If you cut a vein, the blood will just kind of run out continuously. It will not squirt out periodically because it does not have a pulse, right? So in veins, we do not have a pulse. In arteries, you do have a pulse. Example of a good artery is, of course, the aorta. You would do well to memorize the word aorta. Okay, so the aorta is this large, thick artery on the top of the heart. And when blood leaves the left ventricle, it shoots out through the aorta at tremendous pressure. Some of you might be interested in going into law enforcement. A lot of the students from Columbia College graduate and they join the police, or they work as corrections officers, or they try to get into the RCMP. A few Columbia College graduates have gone on to join the RCMP. And if you're in law enforcement, if you're a detective and you're investigating murder scenes, one of the things that you look for is you look for something called aortic spatters. So a spatter is a splash of liquid, right? Now if you cut the aorta, like if a bullet travels through the aorta, or if it's cut with a knife during a murder or something like that, the blood will spurt out, will come shooting out of the aorta with such force that it will travel probably 1 or 2 meters across the room. So if you can find the aortic spatter, if you find somebody was murdered with a knife or something like that, if you look for the aortic spatter, you can tell which direction they were facing and in which part of the room they were facing when they were stabbed. Okay, so aortic spatter is something that you can look for only because of the fact that blood travels through the aorta with such great force. Okay, and the other types of arteries that you might want to memorize is the the coronary arteries. The coronary arteries, of course, are the ones that take blood to the heart itself, and so they are very important, because if the blood doesn't get any heart, then if the heart doesn't get any blood, then the heart stops and you die. You know, there are other parts of the body where you can have blood cut off. You can cut off the flow of blood. By the way, when you cut off the flow of blood, the term for that is called ischemia. You can have ischemia to any other part of the body except for the brain, and it wouldn't kill you. But if you cut off the blood supply to the heart or to the brain, it would. Okay, capillaries, they are organized into networks called capillary beds. Capillary beds. They are microscopic, and they supply oxygen and nutrients to the body tissues, as well as absorb the carbon dioxide and waste products and take it away. So these nutrients and gases are meant to be exchanged through the walls of capillaries, and that's why the capillaries have such thin walls. The wall is made of endothelium, which is just a fancier name. It's another name for squamous cell epithelium that happens to be found inside a blood vessel. Okay, here is a light microscope image of a capillary. You can see how thin it is. Those red things inside it are erythrocytes, or red blood cells. And you can see that the capillary is just wide enough to let the red blood cells flow through it in single file, in single line. All right, and do you remember at the beginning of the course I told you approximately how big a human cell is? So a human cell, on average, is about 10 microns in diameter. It's about 10 micrometers across. And so you can see that this blood vessels, red blood vessels generally are a little bit smaller than regular cells. They tend to be about 7 microns. So you can see that this capillary is about 6 or 7 microns in diameter. So that's very, very small. It's just big enough. It's just big enough to allow one red blood cell through at a time. Right? That's very, very small. And these capillaries are usually organized into net-like structures because you can cover more. They're meant to cover the whole surface of an organ or cover the whole surface of a tissue in order to supply oxygen and etc., nutrients and oxygen to the whole organ. All right, so in our diagram here, this is meant to be a capillary. You can see it's an extensive network of very thin capillaries. Okay, veins, their job is to bring blood, oxygen-depleted blood, back to the heart, but as you saw, there are some notable exceptions around the heart where you call the pulmonary veins, for instance, are carrying oxygen-enriched blood from the lungs back to the heart. So there are some exceptions to that rule, but generally veins carry oxygen-depleted blood back to the heart at low pressure. They have valves to prevent, they have a thin layer of connective tissue and smooth muscle because they don't really need to contract. Veins don't contract. You don't have vein dilation or constriction. That's usually something that just arteries do. Blood is traveling through them at low pressure, so they don't need to be reinforced by an extra layer of connective tissue for extra strength. They have valves to prevent blood flow in the wrong direction. They do not have a pulse. They do not have a pulse. Good example of, also I should mention that muscle movement helps circulation through veins, and I'll show you what that means in a minute. So a good example of a vein to memorize would be the inferior vena cava. Do you remember when we discussed the digestive system, we talked about the hepatic portal veins and the hepatic portal system and the hepatic portal vein itself? The hepatic portal vein is the large vein that carries blood, nutrient-enriched blood from the intestine, where we've digested our food, to the liver, where the toxic compounds that are in the digested food can be removed. Right? And then, so blood that contains digested food must go through a filter first, and that filter, of course, is the liver. Okay, so here are some, this diagram shows us where some of the important veins are. The inferior vena cava is this, all the way down here, right? And the inferior vena cava travels all the way up from the bottom of the body. It carries the oxygen-depleted blood back from the organs as well as the legs. It does not take blood directly from the intestine. the blood has to first pass through the liver. So the hepatic portal vein takes the blood from the intestines to the liver in order to filter out the poisons. Then the blood goes from the liver to the inferior vena cava through another vein called the hepatic vein. And I can almost guarantee that I will try to trick you on a test by asking you some question where you have to distinguish between the hepatic portal vein and the hepatic vein. Right, so the hepatic portal vein goes from the intestines to the liver. The hepatic vein goes from the liver to the inferior vena cava. Okay, now we mentioned the fact that movement of muscles aids the flow of blood through veins. So the blood is flowing through the veins so at such low pressure that it will often stop. Unfortunately, if the blood stops, it will start to clot. Right, it will start to clot, and if you have blood clots in your veins, because the blood is, it's not good for the blood to be standing still. If the blood stands still, it will start to clump together and form clots. That is actually referred to as deep vein thrombosis, and it's very bad for you. It can cause, if those clots circulate around the body, they could clog the coronary arteries, for instance, and give you a heart attack. So you want to avoid having the blood stop. But the blood is traveling so slowly through the veins that sometimes it does stop. Now we have these valves, but how do they work? How do valves work if the blood has stopped altogether? Well, here you see on either side of a vein, we have a muscle on either side of the vein. Let's pretend that these are leg muscles. And then you have this big vein running up through your leg. You want to make sure that the blood goes upward through the leg and not downward or not stopping altogether. So when you move your leg muscles, when you get up and walk around, that flexes the muscles in the legs. When you flex the muscles, they get bigger in the middle, and that has the effect of squeezing this vein, right? So we squeeze this vein. Normally, if you squeezed it, the blood would flow in both directions, wouldn't it? To get away from the area that you squeezed, the blood would flow in both directions, except that if the blood flows in the wrong direction, it causes the valve below the squeezing point to slam shut. Causes the valve that's pointing in the wrong direction to slam shut. And the valve that's pointing in the correct direction will open and allow the blood to go through. So you can see that if you have poor blood circulation, the best thing you can do to increase the circulation is to walk around and to move around and to move a lot. So you'll see that a lot of elderly people have poor blood circulation. If they sit down too long or they stand in the same position too long, their feet will begin to swell up because the blood is collecting in the lower points of the body. And the way to get the blood to circulate and move around is to, if possible, to get up and move around, right? To get up and move around. And the getting up and walking around and doing exercises and so on causes the muscles in the legs and the arms to contract and squeeze the blood vessels, and squeezing the blood vessels causes the blood to flow in the right direction only, and the flowing in the right direction only is something that's facilitated by these valves. Okay, let's learn some of the names of the major arteries. You do need to know these. I might ask you some of these names on the test. Right, so the aorta, of course, runs through the thorax, and it is the first blood vessel to leave the heart. The subclavian arteries, do you remember when we learned about the clavicle? So the term clavian refers to the clavicle. So the subclavian arteries flow under the collarbone and connect with other arteries that flow down the arms. So from the aorta, we have the subclavian veins that run underneath the clavicle and then down the arms. The carotid arteries flow up the neck to the head. There is an artery called the common carotid artery that flows up the side of your neck. Right? So that's very important because it takes blood to the brain. The brachial arteries are the arteries that are right in your arm. They're in the upper part of your arm. And when you measure blood pressure, which we're going to learn about in a minute, you measure blood pressure using the brachial artery. That is the most common artery to use to measure blood pressure, right? So that's a question I'm almost certain to ask on the test. Which artery do you use to measure blood pressure, for example? Right. Okay, the femoral arteries run alongside the femur, which of course is the bone in the upper leg. Then we have a hepatic artery. the hepatic artery travels from, supplies blood to the liver. In order to be a filter, the liver does a lot of work, and if it's going to do a lot of work, it needs a lot of fresh oxygenated blood. So the artery that takes blood to the liver is called the hepatic artery. The artery that takes blood to the stomach is called the gastric artery. The artery that takes the blood to the kidneys are called the renal arteries. There's a left and a right renal artery because we have a left and a right kidney. So those are names you should know. Right, so here is the subclavian artery is the red thing that's visible behind the blue thing, and then the brachial artery is located here, this large artery that's running down the upper arm. The aorta is up here, and then you have the left and right renal arteries down here, sort of. The left and right femoral arteries are running down the legs here. All right, let's learn the names of some of the major veins. You know about the superior vena cava and the inferior vena cava. You know about the jugular veins. The jugular veins bring blood back from the head down to the heart and then ultimately put back into the heart through the superior vena cava. The subclavian veins bring blood back from the arms, right? So they run they run underneath the clavicle. Okay, here's an important vein that you must know the name of. The median cubital vein is located right above your elbow on the inner surface on the anterior side of your arm, just above the elbow. It is the vein that is usually used to take a blood sample, right? So if I ask you which vein is usually used to take a blood sample, you would answer the median cubital vein. Right, so that is definitely one you need to know. And then we have the hepatic portal vein, which takes blood from the intestine to the liver. That is different than the hepatic vein, which takes blood from the liver to the inferior vena cava and puts the nutrient-rich blood back into circulation. Then there's a gastric vein. There are a left and right renal vein for the two kidneys and so on. So these are names you should be familiar with, especially the median cubital vein. Right. Now, just to give you another little bit of career advice, if you're interested in working in hospitals and things like that, and you're interested in that, if you want a profession, if you want to get a job in one of what are called the allied health professions, so doctors and nurses, they say those are the medical practitioners, and then there's a bunch of other people that are sort of in orbit around doctors and nurses that do jobs in the hospital that are related to what the doctors and nurses do, and we call those allied health professionals, and that includes physiotherapists, lab technicians, and so on. So if there is a job that you can get, one of the shortest training courses that you can take at BCIT, the British Columbia Institute of Technology, is their course in phlebotomy. The word phlebotomy, the word phlegm, is an ancient word meaning the blood. Right? So a phlebotomist is somebody whose job it is to take the blood samples. Right? So you go to the doctor says, I want you to take a blood sample, and they'll send it to the lab and analyze it. Right? So you go to another room and somebody takes a blood sample out of you, but that is not a doctor or a nurse who took the blood sample. It's usually a phlebotomist. So a phlebotomist is somebody who spends about 2 months taking a course at BCIT, and they learn just how to take blood from every possible vein and every possible artery in the body, and then they give you a certificate. And with that certificate, you're allowed to apply for jobs in the hospitals. I was trained as a phlebotomist, so I know how to do it, but I did not take the BCIT course. I took it as part of something else that I had to do for a research job I had once in a while. So I can do phlebotomy, but I don't have the certification, and therefore I could not be hired by a hospital to do that. It's illegal, right? The hospital, by law, is only allowed to hire certified phlebotomists to do that kind of work. Right, so the fact that you know how to do it doesn't count. You need to be certified. So phlebotomy is a nice profession if you don't mind poking people with needles all day. That's not something I would want to do for a living. I wouldn't want to do that for a living because I don't like hurting people. I worked one time in a research lab in the Children's Hospital, the BC Children's Hospital, which is in Vancouver. And the lab was next door to a room where they took blood samples from babies all day long. So the technicians who worked in this lab were miserable because all day long they would hear the babies screaming in the room next door because the babies don't like to be poked in a heel with a needle. So that makes the babies cry. So the technicians who were working in this research lab were depressed because they had to hear babies crying all day. Just imagine how the phlebotomist felt. you know, that the phlebotomist, either you would become desensitized to that, or you would look for another job. So it can be a good job depending on what kind of person you are. It can be a good job, it can be an unpleasant job. Either way, it doesn't take you very long to get your qualification as a phlebotomist. Now, when they send the blood sample to the lab, it's analyzed by somebody who is a certified medical laboratory technician, MLT. And the certified MLT program is also taught by BCIT, and it takes 2 years. Right? So you finish your AD at Columbia College, or you could even transfer after first year Columbia College. If you have permanent resident status, you go to BCIT and you can get a job, or you go to BCIT and you can be trained as a medical laboratory technician, a CMLT. The CMLT training includes phlebotomy training. So if you're a certified medical laboratory technician, you know how to do all the tests on blood samples, on urine samples, on stool samples. You know how to do all the tests for microbiology tests to analyze infections. And in addition to all that, you have also been trained how to do the phlebotomy. So the phlebotomist who takes your blood in the hospital might be just a phlebotomist, or they might actually be a medical laboratory technician. It's probably better to get if you only want to train for 2 months, you can get certified as a phlebotomist, but your chances of getting a job are better if you do the whole 2 years and become a certified medical laboratory technician. All right, so anyway, a phlebotomist knows that the median cubital vein is the main vein where you would take a blood sample. There are several other veins where you could take a blood sample, but the one that you would go to by default would be the median cubital vein. Okay, so the median cubital vein is right there. It's the one that crosses over the elbow, and it's very easy to spot, very easy to get at. The subclavian veins are here, running under the clavicle. The inferior vena cava is here, right? And then the superior vena cava is there, basically, right above the heart. The jugular veins are right here, running down the neck, and so they bring oxygen-depleted blood back from the brain. And then we have the two left and right renal veins next to the kidneys down here. Okay, let's learn how to measure blood pressure. This method that you've probably seen people do was invented by a Russian physician named Nikolai Korotkov around 1900, and so some of the things that take place in this procedure are named after Korotkov. Okay, so basically what you do is you take the brachial artery, which is located just above your elbow. You wrap you're going to measure the blood pressure of the brachial artery. You take an inflatable rubber cuff called a sphygmomanometer, and you wrap it around the arm just above the elbow. And then you put a stethoscope on top of the brachial artery just below the sphygmomanometer so that you can hear the blood pumping through the brachial artery. Right? And then what you're going to do, when you hear the blood thumping inside, the brachial artery. They call that sound Korotkov sounds in honor of this guy, in honor of Nikolai Korotkov. All right, so what you're going to do is you're going to pump air into that sphygmomanometer, the rubber cuff, the inflatable cuff, and you're going to pump air in there until you have such a great pressure that you can no longer hear the Korotkov sounds. Right? And then you're slowly going to let the pressure out until you start hearing the sounds again. As soon as you start to hear the thumping of the blood inside the artery, the Korotkov sound, you record that as the systolic pressure. And for a normal, young, healthy adult, the systolic pressure is usually about 120 millimeters. Now, air pressure is measured in millimeters of mercury. In other words, you have a tool called a barometer that the pressure of the air forces a bunch of mercury to rise up inside a tube, and you measure how far the mercury rose up in the tube in millimeters, right? So air pressure tends to be measured in millimeters or millibars, right? So you measure the pressure. It's usually 120 millimeters or millibars in the instead of using a barometer, which is what you would use if you were measuring the air pressure for weather, you measure the air pressure that's in the sphygmomanometer cuff, right? So typically the systolic pressure is 120 millimeters. And then you continue to release the pressure until you no longer hear the carot cough sounds anymore. And as soon as you stop to hear the carot cough sounds, you record that pressure as the diastolic pressure. And it is, for a young healthy adult, it's normally about 70 millimeters. And then you report the blood pressure as a ratio of the systolic pressure over the diastolic pressure. So you've heard people say 120 over 70 or 120 over 80, something like that is normal blood pressure for a young healthy adult. For young children, it deviates quite a bit from that. For older people, it tends to be higher than that. You know, for instance, I'm 60 years old, right? My blood pressure typically is around 150, not 120. 150 over 80, not 120 over 70. That's not good. It means that I eat too much salty food, I drink too much coffee, and I don't get enough exercise, and that is a frequent cause of high blood pressure. Constant high blood pressure is, by the way, is called hypertension. Hypertension is another term for high blood pressure. And that's very common in older adults. It's very common for older adults to have hypertension. But there are things you should do to try and, you know, it's not good to have high blood pressure. If you can avoid it, you should, right? So don't eat salty food. Don't drink a lot of coffee. Exercise more to allow the exercising, doing a lot of exercise, 20 minutes, half an hour of exercise a day, cardiovascular exercise, running or pedaling on the bicycle, something like that. It sends a lot of blood through the arteries and the veins and causes them to become more elastic, and the more elastic they are, the lower your blood pressure will be. Okay, so one more time, how do we measure blood pressure? Air pressure is measured in millimeters of mercury, so we measure it using a millimeter. It's actually a barometer, but it's attached to a device called a sphygmomanometer. So you put the sphygmomanometer, it's a cuff, it's a rubber inflatable cuff that you put over the brachial artery just above the elbow. And then you put the stethoscope just below the cuff so that you can hear the thumping of the blood against the walls of the brachial artery. You pump the pressure, you raise the pressure in the cuff until you cut off blood in the brachial artery, and then you will not hear the carot cough sounds anymore. Then you release the pressure slowly, and when you start to hear the carot cough sounds again, when you start hearing the sounds again, you record that pressure as the systolic pressure. As I said, the systolic pressure is usually 120 millimeters for a healthy person. And then you keep releasing it until you can no longer hear the thumping, because the artery is no longer constricted. Like you don't actually hear the blood pumping against the artery if you're not putting any pressure on the artery. So you fill up the sphygmomanometer first until you stop hearing the sounds. You release the pressure, and when you start hearing the sounds again, you record that pressure as the systolic pressure. And then you continue to release, and as soon as you stop hearing the sounds again, you record that as the diastolic pressure. It's usually around 70 if you're healthy. And you report it as a ratio of the systolic pressure over the diastolic pressure. I always manage to trick a few students every year on the test by saying, what is the normal systolic pressure over, what is the normal systolic pressure of a young healthy adult? And they write down 120 over 70, and I say that's wrong. I asked you for the systolic pressure, you gave me the systolic pressure and the diastolic pressure. Or I could say, what's the diastolic pressure of a young healthy adult? Your answer would be 70. If you gave me the answer 120 over 70, that would be wrong, because those are two separate numbers. Okay, this is an illustration. So you wrap the sphygmomanometer around the arm. You pump it full of air, raising the pressure, right? And then when you stop hearing the sounds, you start releasing the pressure again. And when you start hearing the sounds, so you go beyond the point where you stop hearing the sounds, and then you release the pressure a little bit, and as soon as you start to hear the sounds again, you record that number. In this case, it's 120 millimeters. Then you continue to release the pressure until you stop hearing the sounds again, and you record that pressure as the diastole. So that would be the systolic pressure, recording the systolic pressure over the diastolic pressure, and we no longer say millimeters, because if you have 120 millimeters over 70 millimeters, the millimeter units cancel each other, so you just record it as a raw number. And why do you not actually divide it? Right? You actually you actually give me both numbers, because the two numbers tell you different things, right? So the two numbers tell you different things. You could have really low blood pressure, and if both the systolic and the diastolic pressure are low, then you would get the same ratio as you would if both the systolic and diastolic pressure were very high. So you need to know, so you can't just report the ratio, you have to give both numbers, but in the form of a ratio. So you have to show me both numbers. Okay, so this is a sphygmomanometer. So there's the inflatable cuff, and this is actually the barometer here, which is measuring the air pressure in millimeters of mercury. And this, of course, is a stethoscope that you listen to the heart with. Right, so this is how you hook it up. You hook the cuff around the arm just above the elbow, listen to the brachial artery using the stethoscope while you're simultaneously measuring the pressure there. You can now buy in the pharmacist, in the drugstore, in the drugstores these days, you can buy electric blood pressure monitors that take the pressure for you. If you're going to buy one of those, I recommend you buy this one, because this one is meant to measure the blood pressure at the brachial artery. This one here that goes around the wrist is less accurate, because it depends, the pressure that you get will depend on whether your wrist is elevated or lowered, right? So if you're holding your hand up in the air, you'll get an inaccurate number that's too low, because the blood is flowing down your arm due to gravity. If you hold your arm down at your side, you'll get an artificially or an inaccurately high number, because the blood is flowing down your arm with the aid of gravity and causing a higher blood pressure in the hand and the wrist. So the idea behind wrapping the sphygmomanometer cuff around your elbow, just above your elbow, is that it's hard to put it in the wrong level in that case, right? So I recommend the ones that measure the blood pressure at your elbow. If you do use one that's measured at your wrist, you have to make sure that you kind of have your wrist at the same level as your heart, basically, as if you bend your wrist. You can sit down and put your elbow on a table, and then you can measure your blood pressure at your wrist, and it's relatively accurate. You can see that this one, the wrist one here, is even showing 120/80, which is sort of a normal blood pressure. Okay, now let's talk about fluid in the interstitial spaces as a result of blood circulation. Okay, you know what fluid is. Interstitial spaces means the spaces between cells. Okay, there are basically two types of tissue in the body. There is soft tissue and other tissues. Soft tissues are made up of cells that are sort of loosely packed together. A good example of soft tissues would be fat tissue, adipose tissue. Adipose tissues are loosely packed together, and there's room for liquid between the cells, right? So the fluid, the spaces in between the cells, is called the interstitial spaces. And if you have room for fluid inside between the cells, rather than inside, but between the cells, that's called the interstitial spaces. Okay, there are other tissues like the skin which do not have any interstitial spaces. The cells that make up the skin are hooked together very tightly. They're packed together tightly, and there's no room in between them. But adipose tissue is a good example of soft tissue that has room in between cells, which can be filled with various amounts of fluid. Okay, so if fluid can leak out of capillaries and into the interstitial spaces, causing swelling, which we call edema. Likewise, the fluid could leak from the interstitial spaces. It could diffuse back into the capillaries, causing the swelling to be released. Right? So remember that depending on what's going on with the blood and what's going on with the capillaries, the plasma, not the cells, but the plasma, the fluid that's in blood, could leak out of capillaries and fill up the interstitial spaces, causing swelling, or it could go back into the blood and release the swelling. Okay, so soft tissues have lots of space for liquids in between cells. That's called interstitial spaces. The blood plasma, the liquid portion of blood, is capable of leaking out or diffusing out of the capillaries into the interstitial spaces. If that happens, it causes swelling. That swelling is referred to as edema. Right? And if you want to bring it back, if you want to bring that fluid back into the blood, you can do this by increasing the tonicity of the blood. Do you remember in the very beginning when we talked about chemistry and cells and we talked about a hypertonic versus a hypotonic solution? Right? So if you put a cell into a solution that has a higher concentration of salt than inside the cell, water, specifically water, will leak out of the cell into the surrounding environment and the cell will shrink. On the other hand, if you put a cell into a liquid that has a lower concentration of salt in it, water will leak or diffuse into the cell and the cell will swell up. Okay, so what's happening here is that we can cause water to leak out of the blood if the surrounding tissues, the soft tissue, has a higher salt concentration than the blood. But we can cause the water to leak back in if we raise the salt concentration in the blood. Now, salt doesn't have to be sodium chloride. It could be a number of other types of salts. But proteins that are dissolved in the blood also affect the tonicity of the blood, the concentration of the blood, the concentration of solutes in the blood. Right? So as it happens, the liver produces a protein called albumin. It is sometimes called serum albumin. And one of the ways that the body controls how much water is stored in your interstitial spaces versus bringing it back into the blood is by varying the amount of albumin protein that is produced by the liver. So if you increase the amount of protein, albumin protein specifically produced by the liver, that will cause water to diffuse back into the blood from the interstitial spaces and the swelling will go down. On the other hand, if you lower the amount of albumin, the interstitial spaces surrounding the capillaries will cause the water to diffuse out of the capillaries into the interstitial spaces, and then you have edema, swelling. Right? So just remember that you can cause water to leak into the blood or out of the blood by varying the amount of albumin, the amount of serum albumin that is produced by the liver. Okay, so this is an example of edema, right? You have swelling of the feet. This is specifically a type of edema called pitting edema, because if you push your thumb against the swollen part and then you take your thumb away, you leave a pitting edema. Okay, so this is caused by water diffusing out of the blood plasma into the soft tissues, into the interstitial spaces of the soft tissues surrounding the capillaries. All right, now I want to talk to you briefly about something called anaphylaxis or anaphylactic shock, or just referred to as shock. You know that some people are allergic to things like peanuts or bee stings. And you know that if somebody who's allergic to bee stings gets stung by a bee, their face and other tissues will swell up, swelling. Right? And the reason for that is because they have an allergic reaction, and as we'll discuss when we get into the immune system, which is in the next lecture, if you have an allergic reaction to something, one of the things that happens when you have an allergic reaction is that your capillaries become leaky. And the fluids can leak out of the plasma, and even some of the cells can leak out of the capillaries into the soft tissues. Okay, if you have a massive leakage of plasma out of the capillaries into the soft tissues, which is what causes this massive swelling, if you have a large, massive leakage of plasma out of the capillaries into the soft tissues, what will happen is that the blood pressure will drop very low. And if you have a sudden drop in blood pressure, you may faint because your brain or your heart is not getting enough oxygen, because if the blood pressure is too low, the heart has difficulty pumping the blood around to all the areas of the body. And in fact, the hardest place for it to get blood is upwards against gravity up to your head, right? So then you'll probably pass out. You fall on the floor. Now suddenly your head is at the same level as your heart, and you wake up again. But that is referred to as anaphylaxis, where you have an extreme allergic reaction to something like a bee sting or peanut, and you have a rapid drop in blood pressure, and the drop in blood pressure is so dramatic that you may faint, and then if you don't get enough blood to your brain, you'll faint. If you don't get enough blood to your heart, you'll die, right? So it's potentially a lethal reaction. So that's what they say when they say a patient is going into shock. That means that they're having some kind of a reaction to something that causes the plasma to leak out of the capillaries into the soft tissues, and the blood pressure drops dangerously low. Okay, so if you know anybody like that, you know that they carry something called an EpiPen. Right? This is an EpiPen on the right. My niece and nephew are both allergic to peanuts, and so they both carry an EpiPen in case they accidentally eat peanut butter or something. You know, somebody offers them a sandwich, and you go to a restaurant and you ask if this food is peanut free, and they're supposed to tell you the truth. Sometimes they make a mistake because they don't know that there's peanut oil in it, or this particular food was cooked with peanut oil. And so if you eat that and you have a peanut allergy, you'll go into anaphylactic shock. And so the quickest way to save your life is your friend or whoever is with you takes out the EpiPen and they jab it into your leg. An EpiPen usually has an orange side and a blue side, so the saying is blue to the sky, orange to the thigh. So you take the orange cap off, and then you point the orange part into the thigh, and then you slam it into the thigh, and there's a spring-loaded syringe inside the EpiPen that pumps, it goes through your clothing into your leg and pumps adrenaline, which is a hormone, into your leg. The hormone adrenaline will cause your heart rate to increase, and that will bring your blood pressure back up. And it's an emergency way of saving your life. A safer way to save your life is to give you an IV and pump fluids back into your body so that the blood volume in your blood vessels will go back up. But anyway, that's anaphylaxis. And anaphylaxis is an extreme example of what happens to your blood pressure when you have a massive leakage of plasma out of the blood into the soft tissues. All right, let's look at the blood itself and the elements of the blood. Okay, I mentioned earlier on in the lecture that hematocrit is the volume of blood that is taken up by cells. It's typically about 45%. This is how they measure the hematocrit. You take a blood sample out of the median cubital vein, probably. You put it into a test tube, right? So you put the blood into a test tube, and then you spin the blood in a machine called a centrifuge. The centrifuge, actually, if you leave that tube sitting on a bench in a test tube rack for about 8 hours, it will separate into two phases. The blood cells, the red blood cells, will float to the bottom because they're denser than the liquid, and the plasma will sit on top. But that takes about 8 or 10 hours to happen by itself. Whereas if you put the tube into a centrifuge and spin it, you increase the force of gravity on the test tube, and then the cells and the liquid will separate in about 10 minutes. And then you can literally measure how much of the test tube is taken up by the serum versus the blood cells. Okay, now so I said that the volume of blood taken up by blood cells is around 45%, so you can see that in the tube here. And notice that we have red blood cells on the bottom. Those are erythrocytes. And then we have white blood cells sitting directly on top. We sometimes refer to the white blood cells that are sitting on top as the buffy coat. Right? And then we have a yellow liquid in the top of the tube, which is the plasma, which takes up about 55% of the volume. So the erythrocytes are red blood cells. The erythrocytes take up about 45% of the volume. The leukocytes are the white blood cells, and they take up less than 1%. And then the plasma takes up around 45%. Okay, so the buffy coat is made of white blood cells, also known as leukocytes. And look at how many white blood cells there are relative to the number of red blood cells. So you can see that only about 1% of the blood cells are white blood cells. Right? So only about 1% of the cells in the blood are white blood cells. So the red blood cells outnumber the white blood cells by more than 10 to 1. This is what it looks like in reality. Okay, so we have this is what the blood serum, blood plasma looks like this. It's a cloudy yellow liquid. Then we have the white blood cells, the buffy coat sitting in between, and then we have the erythrocytes. Notice that the white blood cells in this tube are obviously more than 1%, and that's because they didn't spin it for very long. But if you actually count the cells, if you do a complete blood cell count, that's called a CBC, you will actually find that only about 1% of the blood cells are leukocytes. Okay, so the erythrocytes are the red blood cells per microliter of blood. Right, a microliter is 1 cubic millimeter, by the way. So that's a drop, basically, right? A microliter of blood is a small, very small quantity, about the size of a drop, a very, very small drop, maybe the size of the head of a pin, right? So that's a microliter. Within each microliter, we have 5 or 6 million erythrocytes, and I will not ask you that exact number. And we have 5,000 to 10,000 leukocytes, and there are several different types of leukocytes that we are going to discuss when we talk about the immune system, which is the next lecture. Notice that there's also 250,000 to 450,000 things called platelets, and this is a good time to introduce platelets. Platelets are what are responsible for blood clots, blood clotting. Right? So the purpose of platelets are sometimes called thrombocytes. And platelets or thrombocytes are used to patch holes in the blood. So have you ever had an automobile tire, a car tire, a very expensive tire that contains a gas inside it, so that if you get a puncture, if you puncture the tire, that liquid, not a gas, but a liquid, there's a special liquid that you can have put inside these very expensive tires, that when they get exposed to the air, it becomes a solid and plugs the hole. You can also buy this, you can buy that stuff in a can, right? So if you have a puncture, you plug this can into your tire, and it's a gas, a liquid spray that you spray into the tire. It pumps up the tire, and the liquid will form a solid where the hole is to fix the puncture. So platelets are sort of like that. They are soluble, but if they run into a hole in one of the blood vessels, they will kind of cluster and plug the gap. So if they do, their normal function is to plug up holes or internal damage to a blood vessel. That's the job of a platelet or a thrombocyte. They can also cause blood clots. So if they form a clot on the inside of a blood vessel to plug a hole, and if that plug is referred to as a thrombus, right? So a bunch of platelets that are plugging up a hole are called a thrombus. So when I mentioned earlier that if your blood stands still, it tends to clot, and it causes a disease called deep vein thrombosis, I meant that the platelets, if they stand still in the blood, they will start to form a clot or a thrombus. Okay, so platelets are there to cause blood to clot. The only reason blood clots is in order to block holes in blood vessels. So that's basically what happens. That's what platelets are for. Okay, so the hematocrit is normally 45% of the blood. Erythrocytes is the technical name for red blood cells. There, after about 5 weeks, I've finally shown you how to spell the word erythrocyte. So erythrocytes are the red blood cells. They are technically dead. They have no nucleus, and they are filled up with the protein called hemoglobin. So when red blood cells are developing, do you remember where they develop? Which type of bone tissue do they develop in? Red blood cells develop within the spongy bone, the calcaneous bone. When they're developing, they're live, and they fill up with They produce lots and lots of hemoglobin, and when they're filled to the top with hemoglobin, they die and the nucleus dissolves. When the nucleus dissolves, it causes the cell to kind of flatten out a little bit so that it's kind of donut shaped. It's the shape of a donut, and that's because the nucleus has dissolved. I mentioned earlier in the course that birds, when we talked about the respiratory system, I told you that birds are particularly energetic because their erythrocytes are not dead the way they are in mammals. Their bird erythrocytes are live, so they last a lot longer. However, in humans, the erythrocytes, and other mammals, the erythrocytes are dead, so they're just dead bags of hemoglobin that are circulating around the body. And they circulate for about 120 days, and then after 120 days, they finally wear out to the point where they get trapped in an organ called the spleen. The spleen is located just to the left of the pancreas. One of the main functions of the spleen is to break down worn out red blood cells and recycle the components. Recycle the iron, recycle the heme, break down the hemoglobin back into amino acids and send them into the blood. Right, okay, so the erythrocytes are dead, they're filled with hemoglobin, they circulate for about 120 days, and eventually they are broken down in the spleen and the components are recycled. Those are the erythrocytes. The leukocytes are white blood cells, and they are part of the immune system, and they are live. And the platelets, which are also called thrombocytes, are little fragments. They're not actually cells. They're tiny little fragments that are fragments of that have broken off from a larger cell, a larger type of cell, and they're used to help patch holes in the blood and also to help the blood clot if there's a big hole in one of the blood vessels. Here's a picture of some blood. These things that are kind of bright have a bright spot in the middle. Those are the red blood cells, the erythrocytes. If you take Biology 130, you'll learn that that bright spot in the middle is called a zone of central pallor because that's where the red blood cell is thinnest. The zone of central pallor the size of the zone of central pallor will tell you whether the blood is healthy or not. I will not ask you about that in this course because this course is meant to be much more elementary than Biology 130. Okay, so these other things, these little things up here, those are thrombocytes. So you can see how small they are. And you can see that the red blood cells are smaller than the white blood cells. So this is a white blood cell, and this is a type of white blood cell called a lymphocyte, which we'll talk about later in the immune system lecture. And this is another type of a white blood cell called a granulocyte, which is again part of the immune system. So they have these two types of white blood cells have different functions. You can tell a lymphocyte from a granulocyte because the lymphocyte has a nice round nucleus. Look at the granulocyte. The granulocyte has an irregularly shaped nucleus that, in this case, it has three little lobes to the nucleus, and we call those cells polymorphonuclei. Polymorphonuclei. Poly means many, morpho means shapes, and then nuclei means nucleus. So polymorphonuclei, PMN cells, as they're abbreviated, are white blood cells that have multiple shapes to their nuclei. So that's something you'll learn a lot more about if you want to take Biology 130. If not, forget about it. I won't ask you about it in this course. Let's move on now to blood types. You know about if you lose blood, you need a blood transplant from a compatible donor. Let's talk about the ABO blood grouping system. So first, you need to know that the immune system, the leukocytes, the immune system will attack and destroy any cells that it perceives as being foreign. These are not your cells. What are they doing here? These cells came from somebody or something else. They must be here to attack your body. I'm going to destroy them. That's what the immune system says. The immune system is particularly good at recognizing foreign proteins. And what the immune system is able to do is able to distinguish your cells from somebody else's cells mainly by the proteins that are on the surface of those cells. So every cell has specific types of proteins on the surface, and Red blood cells are no exception. They have a special type of protein on the surface that are called ceramides. We don't go into that in this course, but they have, let's pretend that it's a protein. A ceramide is not exactly a protein, but it's similar enough that we can call it that for the purposes of a simple course like this. Okay, so the red blood cells have proteins on the surface. Those proteins on the surface are encoded. The main protein on the surface of a red blood cell is encoded by a single gene. Right, by a single gene. Now we have, if you have a gene that exists in the human population, if there are multiple slightly different forms of that gene, they are referred to as an allele. Okay, so an allele is a slightly different version of a gene. Okay, so you know that I mentioned earlier in the course that humans are diploid because we have 2 copies of the genome and we have 2 of each chromosome. So on one of those chromosomes, there is a place where you find the gene that encodes the protein that is on the surface of the red blood cell. And because we have 2 of that chromosome, we can have up to each person can have up to a maximum of 2 different alleles at the same time. There are actually 3 alleles in the human population, but each human can only have a maximum of 2 of them. The 3 alleles are called the A allele, the B allele, and the O allele. Right? So the A allele codes for a protein that is slightly different than the proteins that are present in the B allele. And the O allele is a special version of a gene that we call a null mutation. If you take Biology 234, you'll learn a lot about null mutations. But a null mutation is a gene that has been mutated so that it no longer produces a protein at all. Right? So each human being can have at most 2 of the 3 possible alleles. They might not even have 2, right? So if a person has 2 copies of the A allele, we say that they only have 1 allele. And so they will only produce the A-type protein on the surface of the cell. If somebody has 2 copies of the B allele, we again say they have only the B allele, and so only the B protein will be produced and put onto the surface of the red blood cells. On the other hand, somebody might have 1 of each. They might have 1 A and 1 B, in which case they will put both the A and the B proteins onto the surface of the cell. If somebody has 2 copies of the O allele, they will put no proteins onto the surface of the cell. You see? So each person can have 1 allele or 2. There are 3 in the entire human population. There are 3 of these alleles that are circulating around in the population, but within each individual human being, they can have either 1 of them or 2 of them, but that's it. You can't have 3. All right, so if you have 2 copies of the A allele, you will have the A protein on the surface of your cell. Likewise, if you have the AO, if you have 1 A allele and 1 O allele, the O allele that codes for no protein at all. Right? So, but you still have the A allele, which means that you'll still put A proteins onto the surface of the cell. The same is true for the Bs. If you have 2 copies of the B allele, you'll put just the B proteins on the surface of the cell. If you have 1 B and 1 O, again, you'll be putting Bs on the surface of the cell. Okay, what if you have 1 of each? If you have one of the A's and 1 of the B's, you will put both A's and B's on the surface of the cell. And if you have 2 copies of the O, you will put neither on the surface. All right, so if you take B-type blood, right? If the blood cells have B's on the surface, we call that type B blood. If it has A's on the surface, we call it type A blood. If we have both A and B proteins on the surface, we call it type AB blood. And if we have neither of those proteins on the surface, we call it type O. Okay, so what would happen if you transplanted B blood cells with the B protein on the surface into a person that was type A? That person has A's on the surface of their blood cells, and so their immune system would perceive that the B's are a foreign invader and destroy all those cells. All right, but on the other hand, what would happen if you took B cells, or if you took type B blood cells and put them into a person who had type AB blood? Well, yes, they have type A on their surface, but they also have the B protein on their surface already. So they would not destroy those cells because, I've got the B, so it's not foreign to me. I've got the B protein on the surface of my cell, so that's not foreign to me. Right? Okay, what would happen if you put type O blood into a type A person? Those cells would not be destroyed because it doesn't have the B allele. It doesn't even have the A allele, and it's really the B allele that a type A blood person would react to the B allele, the B protein on the surface. But type O blood cells don't have that. They don't have anything, in fact, so they would not reject it. Not reject that. Okay, so if you have AO, you are blood type A. If you have AA, you're also blood type A. If you have the BO or the BB, you're type B. Right? If you have both, one of each, you are type AB, and if you have neither, you're type O. If you look at the blood that's collected for use in the hospitals, you'll see that they're labeled A, B, and O. They're labeled A, B, AB, and O. There are actually 4 blood types. Okay. Now, let's say that we have a bunch of blood that's been donated by various people with different blood types, and we're going to transplant that blood into a patient who needs it. They were in an automobile accident, or they were undergoing some kind of surgery where they lost a lot of blood. You need to transplant some blood back into that person because they've lost blood. Okay, so here we have a patient who is blood type. What's their blood type? They have the allele, the A allele and the O allele. You tell me what their blood type is. If you guessed blood type A, you're correct. Okay, now what I want to do is over here on the left, we have a bunch of genotypes and blood types for donors. And I want you to tell me if if we transplant blood from any of these donors on the left into the patient on the right, you tell me whether that blood has been accepted or rejected by the patient. Right? So if we transplant blood type A into this patient, will it be accepted or rejected? The answer is it will be accepted. What about if we have a patient who has genotype AA, blood type A? Accepted. Okay, what about B? What about either of these two? Right, that will be rejected quite strongly, because the recipient does not have B proteins on the surface of his blood, so his immune system will destroy those cells. Okay, what if we transplant blood type AB into this patient? Will it be accepted or rejected? Well, it will be rejected because even though we have the A on the surface of these blood cells, we have the B as well. The recipient does not have the B protein on the surface, and so that the patient's immune system will perceive of that as a foreign invading protein and destroy it. What if we transplant blood type O? Blood type O will not be rejected. It will be accepted because it has neither A nor B proteins on the surface. So there's nothing for the patient's immune system to get upset about. Okay, you tell me what's going to happen here. Okay, so blood type, I'll give you a minute to try and figure it out, and then I'll tell you the answer. Okay, this patient, the answer, this patient would accept blood type A. They would reject blood type B. They would reject blood type AB, and they would accept blood type O. Okay, tell me now. Okay, the answer is this patient would reject blood type A. They would accept blood type B. They would reject blood type AB, and they would accept blood type O. All right, what about this patient? Patient is blood type AB. They would accept blood type A. They would accept blood type B. They would accept blood type AB, and they would accept blood type O. Okay, let's try one more. This patient would reject blood type A. They would reject blood type B. They would reject blood type AB, and they would only accept a donation from another blood type O. Did you notice the pattern? A patient with AB blood is classified as a universal recipient, meaning that they can take blood from anybody because they already have all the antigens. They have the A and the B antigen already, so they're not going to reject any other blood as being foreign. A blood type O person is classified as a universal donor, meaning that they can donate blood to anybody because nobody is going to reject their blood. Right? About the most common blood type in the world is type O. About 40% of the world's population. It varies from country to country, but it is around 40% on average of the world's population is type O. If you have blood type O, then your blood is in demand. Your blood is very much welcome at the blood donation center. If you have blood AB, they're not so interested in it because you can only donate that blood to another person who has blood type AB, and blood type AB is the rarest of the blood types. Only a few percentage of the world's population have blood type AB. 40% or so of the world's population have blood type O, and then blood type A and blood type B are also smaller numbers. Okay, now here's an interesting problem. What if a man and a woman decide to have a baby? In the old days, we'd say they get married. They used to, before you could get a marriage license, you had to have a blood test. And the reason for that was because people generally didn't have children until they got married, and so if you were planning to get married, then it was assumed that you would try to have children, and you would have to find out whether you have compatible blood types. Because what happens if the mother is carrying a baby that has an incompatible blood type? Right, so these days, a lot of people don't get married when they have children. That's just the modern world, but we still have to worry about blood type compatibility when a man and a woman have a baby together. So what blood type will the baby be? Okay, so what's going to happen here, right? So these examples that I'm about to give you are a little bit of an oversimplification, because if the baby's blood is incompatible with the mother's immune system, you will get a reaction. However, normally it's not the first baby that's the problem, because the mother has the baby develops inside a membrane called the placenta. The placenta filters out the mother's white blood cells. So the baby's blood and the mother's blood never come into direct contact. So it doesn't matter, the only way that your system, we'll talk about this when we talk about the immune system, but the only way that the immune system can mount a defense is if the foreign cells come into direct contact with the white blood cells in the recipient's body. So in the womb, when the baby is developing inside the placenta, the baby's blood and the mother's blood do not actually mix. Except when the baby is born, there's usually some tearing of the placenta on the way out, and the baby's blood and the mother's blood will mix. The mother will be able to mount an immune system the next time that she has a baby. So you have to worry about whether the mother and the father have compatible blood types, because the mother's blood may destroy the baby's blood if they have an incompatible blood type, but it's usually the second baby that's a problem. The first baby, you usually have the first baby and there are no problems. The second baby may be the problem. Okay, so with that caveat in mind, let's look at some possible problems here. All right, so the father has blood type AO, and the mother has blood type O. Right? So that means that the mother can only donate the O allele. Half the time the father donates the A allele, and half the time he donates his O allele. Okay. Now, what if the baby gets that combination? Will that be a problem? It could be, because the mother doesn't have the A antigen. So the mother's immune system, the second time that this couple creates an AO baby, it could be a problem. So the baby could come out with a blue baby syndrome. So the second type of blue baby syndrome, The first type of blue baby syndrome we learned about when we talked about Tetralogy of Flow. Now the second type of blue baby syndrome is where we have the baby's blood cells, red blood cells, being destroyed by the mother's white blood cells. Okay, so the odds of this couple having a baby that is incompatible with the mother's blood type are 50-50. Right? So, and usually it's only the second baby that's a problem. So this couple would have to have two babies in a row that had AO blood type before it could be something to worry about. Nevertheless, it's nice to know which blood type you have. There's another antigen, by the way, on the surface of cells, red blood cells, that is called rhesus factor, RH factor, and that one is even more critical. If you have a mother who's RH minus and a father who's RH plus, then you have to worry more about blood type compatibility. But we're not, in Biology 130 we talk about the rhesus factor incompatibility, but we don't in this course, so don't worry about it. All right, let's try this. What are the odds that this couple would produce a baby that has an incompatible blood type with the mother? So again, the mother is type O, so she has two O alleles, so she can only donate the O allele to the baby. But half the time the father will be giving the A allele to the baby, and half the time he will donate the B allele to the baby. So that means every time that they conceive a baby, it will be an incompatible blood type. But if they have an A-type baby the first time and a B-type baby the second time, it's not a problem. It's only a problem if they have an A-type baby the first time and a second A-type baby. Okay, how about this one? So the mother is donating the B allele every time, and the father is donating the A allele half the time and the O allele half the time. Okay, so when you get this combination, it will be a problem, but when you get this combination, it will not. Right? So again, 50% of the time, this couple will produce a baby that has an incompatible blood type. Okay, how about this one? I'll leave you to figure it out. I could ask you some questions like this on the test. All right, so that's the end of our lecture. By the way, if you enjoyed the ABO blood group exercise, you'll do problems like that again in Biology 234, the genetics class, because it's an ideal example of using a Punnett square. So it's a type of problem that they like to do in genetics classes. So you do a lot of blood type compatibility as exercises in Biology 234. Okay, so to summarize what we were talking about today, we talked about all the various different parts of the heart, the pulmonary circuit, the systemic circuit, and so on. We learned some of the names of the major blood vessels and the major arteries, and we learned about the blood itself, the formed elements, and the plasma, the hematocrit, and we learned how to do blood typing and learned about compatible versus incompatible blood types when men and women have children. Okay, so that's it for this lecture. The next lecture will be on the immune system, where we do a lot more discussion on the white blood cells.